स्पिकरहरूलाई इन्भाइट जम्मा 8 जना स्पिकरहरू हुनुहुन्छ। उहाँमध्ये हामीले 3 जना स्पिकरहरूलाई इन्भाइट गरेका छौँ। इन्भाइटेड स्पिकरहरूमा डाक्टर मुकुन्द रञ्जित सर फ्रम उहाँ एकाडेमिसियन हुनुहुन्छ नास्को। त्यसरी नै प्रोफेसर डाक्टर विजय पन्त हुनुहुन्छ, उहाँ प्रोफेसर हुनुहुन्छ टियुमा। त्यसरी नै डाक्टर पुनमाया महर्जन हुनुहुन्छ, उहाँ सिनियर साइन्टिस्ट हुनुहुन्छ G++ लाइफ साइन्स कोरियाबाट। त्यसरी नै सञ्जय महतोजी हुनुहुन्छ, उहाँ आश्रा रिसर्च नेपालबाट। त्यसरी नै अनमोल मणि महतोजी हुनुहुन्छ एज ए स्पिकर टियु। त्यसरी नै आदित्य घिमिरेजी हुनुहुन्छ टियुबाट र राजेश कुमारजी हुनुहुन्छ एग्रिकल्चर एन्ड फरेस्ट्री युनिभर्सिटीबाट। त्यसरी नै हाम्रो अन्तिम वक्ताको रूपमा हामीले राखेका छौँ सुनिल मिश्रजीलाई। उहाँ अहिले हुनुहुन्न होला, एकछिनपछि जोइन गर्दै हुनुहुन्छ। म सबैलाई हामी यो सेसन 4 को तर्फबाट म सबैजनालाई हाम्रो सेसनमा स्वागत गर्न चाहन्छु। यहाँहरूलाई स्वागत छ। यसरी नै यो कुरा यसरी हामीले मेन यो यस्को सेसन गर्नुको कारण नेपालमा हामी धेरै बायो रिसर्चेसमा हामी धनी छौँ र यो बायो रिसर्च यसलाई फ्युचर कमर्सियल पर्सपेक्टिभमा कसरी युटिलाइज गर्न सकिन्छ भन्ने कुराको मेन मोटो रहेको छ आज हाम्रो यो सेसनमा। र यो सेसनको लागि चेयर गरिदिनु हुन म बालकुमारी गोलेजियालाई यो फ्लोर हस्तान्तरण गर्छु। धन्यवाद।
धन्यवाद डाक्टर रामहरि दाहाल, हाम्रो यो सेसनको बारेमा विस्तृत परिचय दिनुभएकोमा र हामीलाई यो प्लेटफर्म प्रदान गरिदिनु भएकोमा फर्स्ट NRNA एसिया पेसिफिक रिजनल कन्भेन्सनलाई पनि हामी धन्यवाद दिन चाहन्छौँ। आज कोभिड-19 को कारण विश्व नै आक्रान्त परेको छ, तापनि प्रविधिलाई प्रयोग गर्दै हामीहरू भर्चुअल सम्मेलन मार्फत एक आपसमा जोडिएका छौँ। यस अप्लाइड साइन्स एन्ड बायोटेक्नोलोजी सेसनमा हाम्रो आग्रहलाई स्वीकार गरी आइदिनु भएको हाम्रो आदरणीय इन्भाइटेड स्पिकरज्यूहरूलाई धन्यवाद तथा स्वागतम। त्यस्तै स्पिकरहरूलाई पनि धेरै धेरै स्वागत तथा धन्यवाद। त्यस्तै यहाँ जोडिनु भएका अडियन्सहरूलाई पनि धेरै धन्यवाद दिन चाहन्छु। त्यस्तै हाम्रो यस सम्मेलनमा सहभागी हुनुभएको सम्पूर्ण स्पिकर र श्रोताहरूलाई पनि हार्दिक धन्यवाद तथा स्वागतम। हाम्रा इन्भाइटेड स्पिकरहरूको बारेमा त डाक्टर रामहरि दाहालले अघि भनिसक्नु भएको छ। हामीसँग समय जम्मा 2 घण्टाको समय रहेको छ। त्यस 2 घण्टाको समयभित्र हामीले 3 जना इन्भाइटेड स्पिकर र 5 जना स्पिकरहरूको प्रेजेन्टेसन सघाउनु पनि हुन्छ र त्यसमा केही क्वेसन एन्सरहरू पनि हुन्छन्। ती क्वेसन एन्सरहरूलाई पनि एड्रेस गर्नुहुन्छ। तसर्थ हामीले हाम्रो इन्भाइटेड स्पिकरहरूको लागि 18 मिनटको समय छुट्याएको छौँ। 18 मिनटभित्र उहाँहरूले आफ्नो प्रेजेन्टेसन सकाइदिनुहुनेछ र 2 मिनट चाहिँ क्वेसन एन्सरहरू गऱ्यो भने क्वेसन एन्सरको लागि समय हुनेछ। त्यस्तै स्पिकरहरूको लागि 8 मिनटको समय र 2 मिनट चाहिँ क्वेसन एन्सर भनेर छुट्याएका छौँ। आजको हाम्रो पहिलो अब हामी ढिलो नगरिकन प्रेजेन्टेसन तर्फ नै लाग्नुपर्ने हुन्छ। हाम्रो पहिलो प्रेजेन्टेसन चाहिँ हाम्रो डाक्टर मुकुन्द रञ्जित सरको छ। डाक्टर रञ्जितको बारेमा भन्नुपर्दा उहाँ नेपालको प्रख्यात बायोटेक्नोलोजिस्ट, हर्टिकल्चरिस्ट, पोमोलोजिस्ट र प्लान्ट फिजियोलोजिस्ट हुनुहुन्छ। अहिले उहाँ चाहिँ नास्को एकाडेमिसियन र कान्तिपुर भ्याली कलेजको बायोटेक्नोलोजी डिपार्टमेन्टको एकाडेमिक डिरेक्टरको रूपमा आगत हुनुभएको छ। उहाँले नेपालको बागवानी विशेषतः सुन्तला जातका फलफुलहरू, आलु, अलैँची जस्ता बालीहरूको बायोटेक्नोलोजिकल युज गरेर कसरी चाहिँ सस्टेनेबल डेभलपमेन्टबाट चाहिँ कमर्सियल फार्मतर्फ बारेमा धेरै नै गरेर अनि धेरै राम्रो आउटपुटहरू दिनुभएको छ, जुन चाहिँ नेपाल गभर्मेन्टले पनि धेरै बेनिफिट दिएको छ उहाँको रिसर्चहरूबाट। उहाँ हामी सबैको लागि प्रेरणाको ठुलो स्रोत हुनुहुन्छ। अब म उहाँलाई चाहिँ यो फ्लोर हस्तान्तरण गर्न चाहन्छु। सर प्लिज।
हेलो बालकुमारी, can you hear me?
हजुर।
ल थ्याङ्क यू। मैले चाहिँ मेरो आफ्नो मोनिटरमा आइप्याडमा चलाइरहेको तर केही आएको छैन होइन, म केही पनि कन्ट्रोल गर्न सक्ने अवस्थामा छैन। त्यसकारण मलाई चाहिँ मेरो त्यो स्लाइड नै अहिले म प्रेजेन्टेसन गर्न चाहन्छु है, किन 20 मिनटभित्रमा सिद्धाउनु पर्छ। अब मलाई चाहिँ स्लाइड देखाइदिनुहोस् न प्लिज। मैले चाहिँ गर्न सक्दिनँ।
Is it okay? I have shared the slide of Dr. Mukunda Ranjit.
Okay.
I have a slide.
Thank you very much. Slide मैले पनि देखेँ हैन। right. So I would like to speak in English if you guys don't mind. Is it okay?
Yes, sir. Okay, no problem.
All right. Now, the title of my presentation is Use of Biotechnology for Production of Disease-Free Horticulture Plants in Nepal. Okay, as the title suggests, we are going to talk about how we have been using biotechnology for improvement of horticulture plants, especially crops such as fruits, vegetables, spices, and flowers. So when I do my presentation, I will give example of only these horticulture crops. Okay, next slide, please. Cool. Hello.
Is this okay slide, sir?
Okay, मेरो अर्को slide दिनुस् न.
Second slide.
Dr. Dhiraj slide change गरिदिनु होला.
I'm changing the slide, but which slide you are asking?
Okay.
Just go down. Just go down.
भएन?
म आईटी टिमलाई रिक्वेस्ट गर्दैछु मुकुन्द सरलाई स्लाइड शेयर गर्नको लागि. IT team, could you help with Dr. Mukendra? Can you help?
Hello, slide change भइरहेको छैन, please.
IT team.
सक्यो डाक्टर साहबले slide change गर्नु.
Dr.
भइरहेछ अहिले slide?
छैन.
टिमले काम गरिराखेको छ.
स्लाइड चेन्ज भएको छैन.
मैले यहाँ स्लाइड चाहिँ चेन्ज गरिरहेको छु, तर मेरो स्क्रिनमा भइरहेछ, त्यहाँ किन भइरहेछ यो?
ओके, भयो.
यो तेस्रो चौथो यो चाहिँ अब मलाई
सेकेन्ड स्लाइड. Okay.
This?
Okay,
yeah.
now
Okay.
please time says all right. Now I would like to arrange my presentation in terms of the following sections. First, I would like to introduce what it's all about and what are the main problems that we are facing in horticultural crops in Nepal. So it's the question of problem identification. And then most of the horticultural crops are suffering from diseases. And so the main important point is we have to identify the disease using the biotechnological tools. And then we have to also eliminate these diseases so that the crop productivity will be enhanced. And then we are using a very specific method of biotechnology. It's called plant biotechnology, plant tissue culture technology. And then I will conclude my presentation. Next slide, please. Okay, you guys all know that Nepal is still a very much agricultural country, and the mainstay of development of our country is when we do research and developmental technologies in agriculture biotechnology. So I will not talk about all agricultural crops, only horticultural crops and the main fruit crops, vegetable crops, flower, and spices. And I will try to give examples on what we have been doing on banana, citrus, apples, and fruits. And then on vegetables, we'll take a classical example of potato tissue culture in Nepal, flowers, orchids. And I think, Professor Dr. Bijay Pantu will also help me out when we talk about orchid, biotechnology in Nepal, and spices, mainly on large cardamom, which is a very important, export crop in Nepal, and ginger. So what are the main problems? Most of these horticultural crops are propagated through vegetative means, not by seeds. Of course, rootstocks are made by seeds, but when you do the vegetative propagation, then the mother plant is very important. And the mother plant is, if infected with viral diseases, then it will lead to degeneration and decline. So the main problem, when everywhere in the world, is a lack of disease-free or virus-free plants in horticulture crops. So we have to improve the method of vegetative propagation, especially by tissue culture propagation. And the mother plant has to be removed from the viral diseases. If we continue tissue culture without cleaning our mother stock, then we'll have the acceleration of problems. The problems will be magnified. Next slide, please. Okay, now what are the solutions? Now we have to identify the problem. I've identified the problem. The main solution is, I've already told you, the right diagnosis of diseases. And traditionally, diseases are diagnosed by using what is called biological indexing, because most of the viral diseases have got host plants, preferable host plants, and we need to do what is known as biological indexing. Now, in citrus crop, there are two important graft transmissible diseases named CTV, is citrus tristeza virus, and CGD, which stands for citrus greening disease, now is known as Huanglong bean, is traditionally diagnosed by using biological indicator plants called Mexican lime. Huanglong bean can be diagnosed by using the host plant, Madame Venus sweet orange or Poincaré mandarin. We have been doing that in Nepal for quite many years. And then the traditional way of right diagnosis of these viral diseases is double antibody sandwich ELISA. Now these days you can also do what is called reverse transcriptase PCR, both conventional and real-time PCR. And CTV, we have been diagnosing CTV and the potato diseases, viral diseases like potato virus A, potato virus M, potato virus X, potato leaf roll virus, and potato spindle tuber virus are traditionally diagnosed by using double antibody sandwich ELISA. And for cardamom viruses, we have Chirke, C and F stand for Chirke and Furke, and we have been diagnosing these Chirke Furke viruses by using antibodies of PRSV, which stands for papaya ring spot virus, or you can also use potato virus Y because it belongs to what is known as poti virus. And we are using polymerase chain reaction, both conventional and real-time PCR for the diagnosis of CGD or Huanglong bean. So we have to do not only identification of these viruses correctly, we have to also eliminate these viruses. And what we have been doing so far is using three of the main ways to eliminate viruses, known as thermotherapy, exposing the original plants at higher temperature for a certain period of time. And then we are also doing meristem culture for the elimination of viruses. And then for trees like citrus, apple, we can do micrografting. Next slide, please. Next slide. Okay, these are the biological research and development in organization in Nepal. There are certain public institutions and private institutions. Next slide, please. You see, there are many institutions. Now, in Nepal, in South Asia, Nepal is a very, important country for initiating plant tissue culture. We started tissue culture in the early 80s in Nepal, and protocols for more than hundreds of plant species have been developed, and we have been also doing micropropagation of virus-free potatoes. It was the first kind in Nepal, in South Asia. We have also doing banana, strawberry, citrus stocks, large cardamom, chrysanthemums, and orchids. Next slide, please. Now, there is an example of potato tissue culture for elimination of those 6 potato viruses, one potato spindle tuber virus, right? And what we have been also using is what is known as harvesting microshoots and getting them directly to root in non-sterile sand, and then distributing these shoots, rooted shoots, to the farmers for direct planting into the field. But these are the things that we have been organizing in Nepal. Next slide. So you say this is a very old slide that was done in the 90s, and now we have been able to, produce pre-basic seed, that is completely virus-free potato seeds of Kufri Jyoti, Dizure, and Kufri Sinduri. They are the three major, varieties that are recommended in Nepal. Next slide. All right, next slide, please. All right, here is the protocol for the development of virus-free potato seeds, how pre-basic seeds are made in Nepal. Now this shows that, when you produce potato seeds, you have to go through certain cycle, and basically when they are, multiplied in the field for a few generations, we can expect them to be, infected with virus, and then at the top level, you'll be able to see that potato is harvested. Apical meristem is cultured in liquid media. And then when about 2 to 3 months time, we'll be having a shoot tip well developed from the plant. And then we can expose them for what is known as, double antibody sandwich ELISA for 6 viruses. And when all these viruses are, eliminated, we can do the adaptive multiplication of this virus-free plant list through nodal culture that is shown in the bottom here. And these, once you have multiplied it a certain amount of virus-free plant lists, then we transplant them into insect-proof, greenhouses where, potato tubers are harvested. And you got to do the virus testing when you are growing these inside the glasshouse as well. So you can produce the, little virus-free tubulets called, PBS, and these are distributed to the special seed growers, and they will multiply potato seeds called basic seeds for several generations. Next slide, please. Now, this is a regular protocol for development of a double antibody sandwich ELISA for the detection of different viruses. I think this is very popular for, doing, producing virus-free plants of horticulture crops. And these are basically the first step shows the coating of your, ELISA plate wells with antibody. And then you've got to, extract the sap from the veins of the leaf samples. And then the virus will be, attached there if there is virus. And the next step, you got, after 3 successive washing, you got to use the conjugated or enzyme-linked antibody. And then when there is virus, the enzyme-linked antibody will be also there. And then, its presence can be detected by adding substrate. We have, ES will give rise to the substrate complex, and you have a color reaction that can be detected by, a proper spectrophotometer. So ELISA plate reader is what we use for, routine detection of these viral diseases. Next slide, please. Now this is, this is the room where we will multiply the virus-free potato plant list inside the laboratory. Next slide. And these plants are transplanted into the semi-automatic greenhouses where you can produce hundreds of thousands of tuberless per season. Next slide, please. Yeah, these are the plants, which are all tested against those 6 potato viruses. They're planted and they are ready for, production of PBS inside the greenhouse. Next slide. So these are the, potato tuberlets that are produced, in the, we have been producing these things since 1990s in Nepal. And for this technology developed in Nepal, this is the first. Next slide. Now, this is the banana tissue culture. Now, once they have been eliminated from, 2 bananas, then we will be, using this. Next slide, please. These are the bananas, and once you have enough plants, you can do what is known as sand rooting technique of virus-free plants. And this is example of vegetable culture of citrus rootstock, and this is example of citrus micrografting for elimination of those two graft transmissible diseases, citrus tissue virus and citrus greening disease or Wanglong bean. Next slide. Now, this is the basic symptom of nutrient deficiency, which was mistaken for citrus greening disease. The right diagnosis is in next slide, please. Next slide. Yeah, this is the right symptom of infection with Wanglong bean, and this is another table slide for citrus greening disease. Now you can see the citrus being affected, and this is the main insect vector, which is known as citrusilla. And diaphorenicity is supposed to be the main vector for the transmission of citrus greening disease or Wanglong bean in Asian countries. Next slide, please. Now these are the adult and the instars of diaphorenicity, citrusilla. Next slide. Now, this is the symptom of seeding yellows of citrus tissue virus in Euroca lemon. You can see that when you peel the bark off, you'll be able to see the longitudinal bits, and it's called stem pitting. This is the typical symptom of citrus tissue virus in mandarin and in sweet oranges, etc. Next slide, please. And this CTV is caused by a special aphid. It's called citrus brown aphid. And you can see the adult and nymphs of the aphid. The scientific name is Toxoptera citricida, which is a vector of CTV all over the world. Next slide, please. Now I've shown you, the large cardamom can also be done produce virus-free by meristem culture. This is the step of what we have been doing. In the filter paper bridge, you have a meristem which is about 0.2 to 0.5 millimeter in diameter, can be converted into in vitro plantlet, and you have to also test whether the virus Chirke and Furke have been eliminated by this meristem culture technique. Next slide, please. Okay, now this is diagnosis. In the summary, what we have been doing is by using double antibody sandwich, we have been detecting 6 potato viruses by double antibody sandwich, citrus, CTB, and we can also use Mexican lime as a biological indicator plant. For cardamom, the Chirkin Furke virus have been detected by using pomelo ring spot virus or potato virus Y antibodies. And for biological indexing, we have been using first diagnosis of CGD, Madam Venus sweet orange and Ponkan mandarin, which was used before the application of double NB Sandy ELISA or PCR. Next slide, please. All right, so these are how we have been doing the disease elimination. Potato viruses were eliminated by thermotherapy and apical culture, by potato research program in NARC, and by GREAT, which is my own laboratories. And PBX and PBY were eliminated from 2 Bangladeshi varieties of potato, Petinese and Multa, by our institution here. CTB and CGT were eliminated from mandarin by micrografting by GREAT. Chirke and Furke eliminated from Namsai, Goldsai, and Damarsai by GREAT. Disease-free mother plants of mandarin were identified. Then the signs were collected and grafted on tissue culture rootstock of trifoliate orange and Trias citranges under skin tunnels. And then they have been, we have found that these trees gave rise to fruits in 3 years. And that was mainly done in 2 districts of Nepal, Lamjung and Gorkha. Next slide, please. Now, you see, in conclusion, what I would like to say that zero virus PBS production for doubling of yield of potato. Now the potato yield is about 12 metric tons per hectare. 25 to 30 metric tons per hectare can be achieved if 100% of the potatoes are used, potato seeds are used from PBS seeds. Production of CTV and Wangmin-free grafted plants of mandarin to solve the citrus decline problem in Nepal. Now we have about 2 billion rupees worth of citrus production in Nepal. It can be multiplied by almost 1 trillion rupees worth if we remove these two very serious diseases of citrus in Nepal, where we have tested that it can be done. All right, the production of silken fungi-free large cardamom is also very important because in 2010, we exported 4 billion rupees worth of cardamom. Now that is only 10% of what we have. If we produce virus-free plants, it can be almost 40 billion rupees worth of cardamom can be produced in Nepal alone. And in order to do these things, we have to also produce the antibodies and conjugates for routine diagnosis by ELISA. And we can also produce the banana mojito virus and cardamom cucumber mosaic virus free plants of Malbog banana, which is indigenous variety of banana in Nepal. Okay, thank you very much. And I'm sorry I took a lot of time, almost half an hour. Now, if you have any questions, I would like to, see how it has been arranged. Thank you very much.
महत्त्वपूर्ण प्रेजेन्टेसनको लागि हजुरले टाइमै सकाउनु भएको छ। अब हामी क्वेसनहरू चाहिँ लास्टमा नै लिन्छौँ होला। हजुरको प्रेजेन्टेसनबाट हामीले धेरै इन्फर्मेसनहरू पाएका छौँ। हर्टिकल्चर क्रपहरूमा भएका समस्याहरूलाई औँल्याइदिनु भएको छ र ती समस्याहरूबाट हामीले कसरी तिनीहरूलाई समाधान गर्दै नेपालमा एग्रिकल्चरको रिभोल्युसनको लागि के गर्न सकिन्छ भन्ने बारेमा यहाँले प्रेजेन्टेसन दिनुभएको छ। यो महत्त्वपूर्ण प्रेजेन्टेसनको लागि हजुरलाई धेरै धेरै धन्यवाद।
अब हाम्रो अर्को स्पिकर हुनुहुन्छ मिस्टर सञ्जय महतो। महेन्द्र मोरङ विराटनगरमा लेक्चरर र एग्रिकल्चर क्याम्पस लमजुङमा भिजिटिङ लेक्चररका रूपमा कार्यरत महतो लामो समयदेखि अध्यापन र अनुसन्धानमा संलग्न हुनुहुन्छ। एन्टिबायोटिक र बायोफर्टिलाइजर स्पेसल्ली ती क्षेत्रहरू र युजफुल माइक्रोब्सहरूको डिस्कभरीको लागि उहाँ अनुसन्धानमा क्रियाशील हुनुहुन्छ र उहाँको थुप्रै रिसर्च आर्टिकलहरू पनि यसैसँग सम्बन्धित छ र उनै महतो सरलाई यहाँ प्रस्तुतिको लागि हार्दिक अनुरोध गर्छु। महतो सर प्लिज।
धन्यवाद डाक्टर धीरज। अनि आवाज आउँदै
आइरहेछ, सुनिरहेछ।
Okay, thank you sir. So first of all, I would like to thank NRNA for providing the platform for presenting my work. And let me go towards the work that I have done on Azotobacter and its implication in the field on the wheat plant. So basically, before going to the slides, I would like to give a brief introduction about my work. It was done in the Western Hills of Nepal, Lamjung district. And okay. So basically, with this research, we wanted to understand the findings of Azotobacter nitrogen fixing ability in Western Hills of Nepal, where the temperature is cold enough. So the research were very few. It wasn't in the hills region of Nepal. So we had conducted these things. Okay, basically speaking about Azotobacter. Azotobacter, as we already know, it is a free living bacteria which fixes nitrogen in the soil. And in Nepal, the challenging scenario is about the nitrogen deficiency in the soil. So, and the use of inorganic fertilizer has been diminishing the quality of the soil. To complement those things and increase the productivity, what can be done? That was the big question in front of us. So we tried 3 models. One model was let's use only Azotobacter. And another model was, let's use azotobacter with fertilizer, inorganic fertilizer, NPK, nitrogen, phosphate, and phosphorus, and potassium. And the third model was, let's use it with the farmyard manure. And one model was like, let's mix all of them and see the productivity. So once we as we already know that azotobacter is not only fixes nitrogen, but it also produces growth hormones and growth hormones and certain substances that increase the growth of the plant and the seed germination, and even checks and balances root pathogens. So rhizosporic pathogens, which in terms increases the productivity. So with the understanding that azotobacter does such a work, we developed a methodology for Western Hills, where we use the chemical dose, like inorganic fertilizer dose, 120:80:80 for NPK, which is based on the per hectare. So we calculated it for a pot. Our method basically contained a pot experiment in Lamjung district premises, where we used the pot. Why did we use the pot? The question arises. Question is, the pot, we use the pot for a control condition, so that whatever water we put over there, it doesn't leach out the fertilizer that we put in the pot. So if we do it in ground, it was very tough for us to understand the quantity used by the plant. But in pot, it was well maintained, and we had already made such a provision that no water goes out of the pot. So we already, whatever fertilizer we had kept over there, it was used by the plant, or it remained in the soil. That was the biggest thing that we took into the account for pot experiment. So wheat variety was Gautam, and it was done in Lamjung Krishi campus during the year 16-17. And we had 7 treatments with 3 replications. So T1, T2, T3, T1 was basically our control, and control had only soil in it. And T2 had only wheat plant and only nitrogen, only NPK, inorganic fertilizer. T3 was azotobacter and with seed coated with azotobacter. T4 treatment was azotobacter distributed mixed in the soil. And the 5th treatment was azotobacter and NPK, inorganic fertilizer mixing. Our 6th treatment was azotobacter mixed with 10-ton farmyard manure per hectare. Then our 7th and last treatment was azotobacter, NPK, and farmyard manure mixing. So in control, we did not use any kind of manure or fertilizer. It was purely soil and seed. So for one, how did we calculate that? How many plants we have to sow in the pot? So it was basically done by the thing that in 1 hectare, how many plants can be sown. So with this 18 centimeter into 6 centimeter spacing method, we found out that 9,25,926 plant per hectare can be planted, wheat plant can be planted in the field. So as per our pot size, we took 4 plants. It contained 4 plants, 4 wheat plants in per pot. And for them, what will be the amount of nitrogen requirement? That was an NPK requirement that was also calculated. It was calculated by per hectare, how recommended dose is 120:80:80. So that was divided by 9,000 the plant in per hectare, so 9,25,926, and the requirement for one plant in requirement for one plant was calculated. Similarly, for azotobacter, how much how did we use the azotobacter? So it was 33 into 10 to the power 7 CFU per ml was colony that was used for coating 24 seeds. And how much farmyard manure we use? Then for one plant we used per pot means 4 plant, we used 43.2 gram. It is based on the 10 ton per hectare. So with this, all those calculations are over there, that we use 0.28 gram of MOP, 0.56 gram of DAP, and 0.715 of urea. Now, we used Vitavex at the rate of 2 gram per kg seed for removing the smart food rot. So it was basically to disinfect. Now wheat seeds were soaked, wheat seeds were primarily to sowing, wheat seeds were soaked in hot water at 53 degrees centigrade per 10 minutes. Then it was to remove the blight and smut if it is present over there. So for Azotobacter coating, we used sugar solution 10% slurry, and for irrigation, we used 200 ml water at 2 days interval, which decreased with the time. And weeding was done on 35th days and 55th days. So similarly, after after 113 days, we had harvested the plant. So what were the result? The result was basically in line with the findings that we already had in other studies, but it showed promising result in the Western Hills. The study was not done over there. What it showed that basically we will be talking about, since there is 7 parameters, 7 treatments and lots of parameter, then it will be really confusing that what did we find. So basically my presentation will be focused on combined fertilizer with FYM and azotobacter, biofertilizer, chemical fertilizer, and FYM. And one will be with only NPK, means inorganic fertilizer. And one will be with azotobacter. So let's compare these things, how it goes. Now, azotobacter, we used two methods. One we used, one of the method was coating seed with azotobacter. Another method was in soil, directly putting the azotobacter in soil. So what we found the results very much promising for plant height, root length. So it was T7. T7 means azotobacter along with farm aid manure and NPK, inorganic fertilizer. It was found that plant height, root length, tiller number, panicle number, panicle weight, number of grains, biological yield, everything was increasing. So it had maximum increase was seen in T7 treatment. While we can see parameters with maximum value observed in T2. So what T2 showed the maximum value in, it was in root weight. So root weight was very much high in inorganic fertilizer, only on inorganic fertilizer. Panicle number per plant was high, panicle length was high, total biomass was high. But what was the main thing? That grain yield was very much high, biological yield and grain number per plant. Every economical parameter that we need was very much high in combined result, T7. So now, similarly, in control, and minimum output was seen in T control, which was only with the soil. And obviously, since soil is not supplemented with any sort of fertilizer or biofertilizer or anything else from outside, since it was a control, so the value we must expect that it will be lower. So plant height,
sir, you have very few time. Okay, please.
Okay. So parameters with the lowest value recorded with T3 was T3 means only azotobacter used. So leaf length, root weight, straw weight, biomass, biological yield was lowest in this with this parameter. Now you can see in graph that we have kept over here. It is comparing T7, T5, and T2. T7 means you can see T7 is combining all the fertilizer. T5 is with the chemical, and T5 is chemical and seed inoculation means azotobacter, and T2 is with azotobacter with only NPK means inorganic. Then these results showed that everywhere you can see in final height of plant 79.87. So everywhere the parameter in combined result combined result. Whenever I say combined, you think that FYM, Azotobacter, inorganic fertilizer. Similarly, you can see leaf length, leaf width, tiller number, all these things all these parameters were very high in combined Azotobacter combined with NPK and farmyard manure. With panicle weight, you can see with the number of grains, 38. Similarly, you can see in grain yield is very much high in 1.68 ton per hectare. It was found along with in the case of combined treatment. While with Azotobacter, it was only 1.23 or 1.2. This is the main thing you can see here. Grain yield, what does it show? With Azotobacter, we had only 1.2 ton per hectare, and with NPK, we found out 1.26. But when we combined bacteria and or inorganic fertilizer and farmyard manure, we found it the result is very much promising, 1.68 ton per hectare. So with this, you can see I have kept over the several data in this slides. The basic things were those things. So you what were the real findings? The real findings shows that if we use combined if we combine Azotobacter with farmyard manure and NPK, then we get a 63% increase in the yield than control. So my conclusion regarding this shows that you can see here that if you use Azotobacter with inorganic fertilizer, there will be 43% increase, 43% increase in the yield. But if you use it along with the farmyard manure, there will be 63% increase. If you use only an NPK, there will be 13% increase. So the increase gets productivity increases. So if we want to increase the production, there is no need of only putting supplementing it with the inorganic fertilizer, putting lots of the inorganic fertilizer. What we can design or devise is a system where inorganic fertilizer get combined with the biofertilizer and with the mix of farmyard manure. So farmyard manure can be used as a mix, and it can be used in the field to increase the productivity. So we are not in the compulsion of increasing increasing NPK in the field, putting lots of NPK and destroying the soil microbiota, which ultimately is harmful for the soil health, and ultimately it is decreasing the productivity. So these were my findings and conclusion. If I have taken lots of extra time, then I can only thank you for giving such a platform to present my work. Thank you, NRNA.
Okay, obviously Azotobacter is a valuable microbial acid which can promote or compensate the need of chemical fertilizer in our country, as we have the scarcity of chemical fertilizer since recently we heard the several news in the news channel. Thank you, Sanjay sir, for your nice presentations.
Thank you everybody.
अब म ढिला नगरिकन प्रोफेसर विजय पन्तलाई स्वागत गर्न चाहन्छु। प्रोफेसर विजय पन्तले 1992 मा बोटनीमा ट्युबाट मास्टर्स गर्नुभएको थियो र त्यसरी नै हिरोसिमा युनिभर्सिटी जापानबाट बायोटेक्नोलोजीमा 1997 मा पीएचडी गर्नुभएको थियो। अहिले उहाँ सेन्ट्रल डिपार्टमेन्ट अफ बोटनी टियुमा प्रोफेसर हुनुहुन्छ। जनरली उहाँलाई अर्किड स्पेशलिस्ट भनेर पनि चिनिन्छ। उहाँले अर्किड बायोडाइभर्सिटी, यसको अनुसन्धान र कन्जर्भेसनमा उहाँको ठुलो योगदान रहेको छ र यस सम्बन्धी उहाँको धेरै रिसर्च पेपरहरू पनि पब्लिस भएको छ। मलाई लाग्छ उहाँले 100 भन्दा धेरै जर्नलहरू राष्ट्रिय तथा अन्तर्राष्ट्रिय पब्लिकेसनमा पब्लिस गरिसक्नु भएको छ। त्यसरी नै 3 वटा भन्दा बढी किताब लेखिसक्नु भएको छ। त्यसरी नै उहाँले मोर देन 70 स्टुडेन्ट्सलाई सुपरभाइज गरिसक्नु भएको छ, मास्टर स्टुडेन्ट्स। सिमिलरली मोर देन 6 जना भन्दा धेरै विद्यार्थीलाई पीएचडीमा सुपरभाइज गरिरहनु भएको छ। ढिला नगरिकन उहाँले हाम्रो आतिथ्यलाई स्वीकारेर आइदिनु भएकोमा हामी उहाँलाई धेरै धन्यवाद दिन चाहन्छौँ। यसरी नै हामीले यसको सेसनमा उहाँको छोटो प्रेजेन्टेसनको लागि इन विट्रो टेक्नोलोजी फर मेडिसिनल प्लान्ट्स देयर प्रडक्ट एन्ड बियोन्ड टपिकमा रहेर आफ्नो प्रस्तुतिकरण दिनको लागि म अनुरोध गर्न चाहन्छु। धन्यवाद।
थ्याङ्क यू रामहरि भाइ। म स्क्रिन सेयर गरौँ होला हैन?
हजुर हजुर
भयो होला
हजुरको जीमेल देखिरहेको छ अर्को पीपीटी स्लाइड शेयर गर्नुभयो हुन्थ्यो
भयो देख्नुभयो स्लाइड सो भयो
हजुर त्यसलाई फुल स्क्रिन गरिदिनु भयो भने
म गर्छु हस् थ्याङ्क यू भेरी मच थ्याङ्क यू NRNA it's very good to know you all see you all म आज अलिकति टेक्निकल त्यसको एउटा डेटाबेस प्रेजेन्टेसन भन्दा पनि अलिकति ओभरअल नै कुरा गरौँ होला सो why tissue culture is very much important in current scenario for medicinal plants because at this moment when the world is looking for some very good drugs or medicine for COVID, so tissue culture equally important for medicinal plant. I'm going to talk about this. So probably we don't know when the use of medicinal plant started, but it might have started with the human need, and probably 60,000 years ago evidences shows that medicinal plants were there and the people were aware of the medicinal plants. These are some of the pictures. We all are very familiar with these plants, which are tremendously used in various purposes, Artemisia, ephedra, or ginkgo, some, and some more, Catranthus rosas, taxus, aloe vera, atropas, so many, and there are many more. So these are globally used or globally concerned medicinal plants, and there are many in the list actually. And most of the species that we call threatened are of medicinal value. They have been used for this or that purpose. That's why most of the species that we mentioned threatened or some are going to be extinct are, of course, the medicinal plant which has been directly collected from the wild. And I have shown some of the reasons, for example, habitat destruction, modification of the habitat, or fragmentation, means utility of that hotspot area of medicinal plant for various purposes. And of course, many pharmaceutical companies are involved in over collection, and sometimes local people are concerned in the consumption. And we have very destructive harvesting technique. We don't think for next year's product. So this is also one of the reasons. And we have very limited on systematic cultivation practice, as well as we don't have much more techniques for the industrial production of the phytochemicals, which also one of the hindering factors. And these are some of the global slide. I have taken it from internet. This is from Madagascar. You see the market of medicinal plants. Similarly, bark is taken away in many woody plants, which are really difficult to regenerate in nature. So this is also very unsystematic harvesting techniques. And if it is happening globally, not only of our Asia Pacific region or South America or wherever, all over the continent, the alternative means is needed for protection or sustainable utilization of medicinal plants. And that's why Dr. Ranjit already talked about the importance of tissue culture. I'm talking about the same. So this is very useful for sustainable utilization and to produce the product in lab, and we can optimize the condition for the desirable product in lab. So this is from now on, probably my presentation is more pictorial, which must be very enjoyable to you. These all are our slides. We are working on commercial production of different plant species, and this is the slide of orchids. As you have already mentioned, I'm working on my team actually. There are young stars who are working in commercial production of orchids. So why tissue culture? It's not only mass production, but or conservation. We can reduce the cost, we can get the controlled supply of biochemicals, even we can detoxify the metabolites in culture, we can produce under stress, and even we can produce the desirable structure, say fairy roots, or only the what do you say, leaf sections. So there are so many benefits, and this is extraction process is very easy in the tissue culture regenerated plant. So this is important in many aspects. And since there are many issues in the genetic transformation, in this section we are not doing the gene transfer techniques. So this is also very good point. And these are some of the examples that I have just given as example. Say commercial production of ginseng, ginsenoids. It is almost in the lab in Korea. Similarly, some berberine, this is commercially produced in Japan. Morphine, codeine, taxol. There are so many other phytochemical that has been produced in culture in many developed countries for commercial production of metabolites. So I have taken this picture, Takanori, from the Color Atlas of Medicinal Plants in Japan. You can see these roots from Panax ginseng. These all are produced in lab. So you can see this tank, which are commercially this root producing bioreactors. Similarly, nowadays I have given the YouTube link here. The nowadays milking of root, just production of the most of the medicinal plants we use are they produce the metabolites in root. So root milking without any other media, but just supplement the nutrient. That is called milking root. This milking root technology has been very popularized. Similarly, we use colchicine to produce the to induce the proidy, so that more metabolite can be produced in culture. So there are various technology in tissue culture have been introduced to synthesize the metabolite in culture. So I'm going to show some of our work. Actually, my students have done this kind of work. Some example, for example, I'm showing this slide of Oroxylum indicum, which is very important tree in context of our country. And this is a tree species we call Totelo in Nepali, and multi-purpose tree, and it has been put as a vulnerable red in IUCN category. So important, it's very difficult to propagate woody species. One of my students, Ganesh, did this job very nicely. This tree, it has got so many medicinal properties, as well as, I'm not going to talk in detail. So we have been successful to produce this plant, and this is very interesting achievement for us. Similarly, this Valeriana jatamosi, one of my PhD student is still doing different kind of research, but this work has been done by my past student. This is also highly traded, highly desirable species, and most of this its roots are used as a leptorite, anti-leprosic drug, as well as essential oil, very high value essential oil. So Bosant did, actually 3 generations of students did this research. Some did micropropagation, some did its acclimatization process. But our goal was to extract the valuable metabolites, and Basant actually did this job. We produced lots of hairy roots, you see, and we compare this hairy root with all the cultivated species roots, and it was a high concentration of essential oil. I collaborated with chemistry department. So this hairy root contain more baleptorite and other essential oils, all the alkaloid even in this plant in our hairy root culture. So this is also alternative means for metabolite production. So this is ashwagandha. It's interesting work. You can see the roots here. We ashwagandha, Widenia somnifera, and Rauwolfia serpentina. You can see these two different structure here. This is callus as well as root we induced in culture. This is multiple root you can see. These are actually colchicine treated root. So the objective was to induce a high level of ploidy in callus, so that we wanted to have the more metabolite than in the normal plant or normal callus. That's why it was also interesting. You can see the chromosome slide. This is not very clear here, but the phenotypic variation you can see in the colchicine treated plant. This was high ploidy, twice n equal to 44. This is twice n equal to 22. So this morphological variation associated with the metabolite production also, and in callus also we found very good what do you say, concentration of metabolites in the callus. These are all callus we compare with the plant of from different localities. You can say, I think the Sudeep did this work, and from 4 different localities of Nepal we brought the rootstock and compared with the callus, and we found this ploidy induced callus gave the highest concentration of alkaloid in this plant. So it is possible to make the metabolite in lab. These are these pictures. So I must talk about orchids. These are beautiful flower. Global market is very high, one of the top 10 cut flower. But and people are popularizing the orchid differently. You can see this slide. Singapore Botanical Garden is naming whoever dignitaries visit the garden. They give the name for orchid for those person. There are many such plant, and these plants are popularized globally. यसरी नै बनाउनुहुन्छ उहाँहरूले. नेल्सन मण्डेलाको नाउँको स्लाइड हो यो. यो चाहिँ अफ्रिकन अर्किडलाई उहाँहरूले यसरी दिनुभयो. त्यस्तै मदर टेरेसाको नाउँको अर्किड हो यो. This is Dendrobium, our Asian orchids. So I mean, people are doing so many यस्तै गरेर उहाँहरूले आफ्नो अर्किडलाई कसरी पपुलराइज गर्ने भनेर गर्नुभएको छ. You can see this is wine production in Taiwan from orchids. Vanilla, we all love vanilla ice cream. This is South African orchids, so you can see. And there are, this is probably the one of the oldest tea culture started from orchid from China. So these are all herbal tonic and TCM in Chinese traditional medicine. Similarly, you can see all these Ayurveda or Arabian medicine everywhere. The use of orchid is very high. And यो हनुमानको मैले फोटो देखाएको चाहिँ सञ्जीवनी बुटी चाहिँ नि इन्डियनहरूले एकदम हेर्नुभयो भने देख्नुहुन्छ तपाईहरुले. सञ्जीवनी बुटीको वान अफ द इन्ग्रेडिएन्ट्स चाहिँ अर्किड, यसलाई फ्लिकेन्जेरिया भन्छ. र यो स्टोरी रामायणको स्टोरीमा भएको भएर मैले यहाँ देखाएको. सो यो स्टोरी भनेको त्यति बेलादेखि पनि अर्किडको चाहिँ मेडिसिनमा युज रहेछ भन्ने एउटा हाइपोथेसिसकै कुरा भनौं. र यो बेसमा हामीले एउटा बुक पनि लेखेका छौं, मेडिसिनल अर्किड्स अफ नेपाल भनेर. यो अमेजनमा पनि राखेको छ. र आफ्टर द्याट अनि एकजना हाम्रो एसियन अर्किडको चाहिँ बुक आछ. र वी हेभ मेनी सच मेडिसिनल अर्किड्स हियर. यो 5 औँले भन्छौं हामी. यसको जरा 5 औँले जस्तै हुन्छ. र यो दिस इज रियली CITES included plant, but its collection is so illegal and high. Only few spot are left nowadays. किन यसको रुट नै उखाल्ने हो अब मान्छेहरुको दाल भातको कुरा हो र यसको प्राइस पनि हाई छ त्यही भएर यस्तो बिरुवाहरु धेरै यसरी you can see the people sell in tons, not in the few. There are different kind of sellers, but these sacks are all orchid sacks, dried illegal collection. So हाम्रो बुकमा हामीले यो ग्राफ हेर्नुभयो भने most of the paths are underground paths. So people uproot the plant. So the यति 10 वर्ष लागेको यो म्याच्योर बिरुवाको तुरुन्तै अप्रुट भएपछि डेफिनेटली यो होल अर्किडेसी फ्यामिलीलाई साइटिसमै राखेको छ ग्लोबल्ली त्यही भएर त्यो कारण चाहिँ त्यही नै हो इलिगल ट्रेड इज भेरी हाई सो अब यसको सीड जर्मिनेसन हुन पनि एकदम गाह्रो छ because there is no endosperm in this plant. So only 2% seed germinate in nature. That's also when they come in contact with the fungal partner and the fungus work for the carbohydrate supplement and then the germination process start. So the alternative is only tissue culture. So we have been doing lots of work on tissue culture. मेरो time कति छ होला?
Continue गर्नुभयो हुन्छ
हजुर
Continue गर्दै गर्नुस् न म इन्फर्म गर्नेछु
Okay, so we have already done about 40 species of orchid has actually been conserved in our lab. Unfortunately, we lost many species during earthquake, but we are still again continuing the same work. So you can see this is very interesting structure protocom that is only found in orchids. So we are doing protocom culture in some species where it responds very good, and this mass production process can be done very efficiently. In some species, it won't work. So we apply, as Dr. Ranjit already suggested, meristem or suchip culture. And I have worked with Dr. Ranjit. We have worked on virus-free orchid production. We worked together. It was interesting finding again. So in this ground orchid, we did, in some ground orchid, we are doing the suchip culture or meristem culture technique. In some plants, we have found the aerial roots are very interesting structure. You can see the plant multiplication from aerial roots. Now we have done a very interesting work on artificial seed production. So there are many vegetative propagating plants. We do not bear seeds. So this is one of my PhD students' work, Sriti. She did very nice work on artificial production in orchids. We have tried in many species. She had been successful in this species. And you can see from a single seed, we can produce millions, thousands. You can see this bunch of buds within 4 weeks or something like that. So those plants which take 8 to 10 years to mature or to grow, you can produce within 6 months in lab. So this is fantastic finding actually in this species. And we are taking this species as a model plant for other research also. And you can see this germinating artificial seed in substrate. Very interesting. It's coming up. And we have tried various substrates. So I'm trying to work with commercial grower in this aspect so that they are more good researcher than us in terms of plant growth. So we have to collaborate with the commercial grower. And one of my PhD students, he's working on fungus isolation from the orchids, which are really boosting the mother plant to be resistant in adverse condition. Must be they are providing the medicinal compound. And we tested the, growth promoting effect of this some identified fungus and their extract on growth and promotion. So this is Sujit's work. He has done very marvelous work in this growth promoting effect. He had done this work even if fungus colonize and plant can grow together or not. This is electron microscopic photographs of Sujit. He did this work in Pune, India. We had a collaboration. So this fungus actually help to orchid to survive in adverse environment, and we are going to check for their role in anti-cancer assay also in our next project. So these are some of the interesting acclimatized plants from different species. Student excited to show these plants. They could make the plants survive outside. And this is my commercial production. You can see small poly bag. Actually, the Korean government has supported us. I'm collaborating with Annapurna Research Center. So these are all, what do you say, ready plant for the transplantation. And we are going to transplant this commercially produced plant in rural people, so that people, stop illegal collection and grow plant, get benefit, and it will definitely help in the economy also. And we have, one of my students is, our main goal from orchid, I have shown some micropropagation or some interesting tissue culture technique, but our main now project is we are trying to extract the anti-cancer agent from orchids. There are various literature, and there are various literature that some of the extract have already proven to be anti-cancerous in lung cancer, ovarian cancer, or other different kind of cancerous cell. And even Taiwan and China, they are providing the TCM for anti-cancer therapy. So we are trying to get such compound in our lab. We are not extracting from the whole plant, but we are developing some kind of tissue. You can see various kind of tissue, various kind of media that can proliferate this tissue, and from which we will extract the anti-cancer compound. And success has been achieved in some extent. We have done MTT assay. Pushparaj, this is my PhD student, is working on anti-cancer assay of some of the orchid species, and you can see these are HeLa cell line, and testing with the control and orchid extract. So having said so, I'm not म यहाँ अब तपाईहरुलाई सबै डिस्कसन रिजल्टको डेटा अहिले अब यो सर्ट समयमा म बताउन अलिकति सक्दिनँ होला, तर मैले सेयर गरेको कुरा चाहिँ इट्स सो इम्पोर्टेन्ट टु अहिलेको समयमा अब यो जङ्गलमा गएर बिरुवा बायोडाइभर्सिटी कन्जर्भेसनको कुरा हामीले हाम्रो रेस्पोन्सिबिलिटी पुरा गर्ने हो भने त्यो बिरुवाको मेटाबोलाइट ल्याबमा बनाउन सकिन्छ, अथवा त्यो बिरुवालाई मास प्रोडक्सन गरेर कल्टिभेट गरेर पनि हामी त्यो सप्लिमेन्ट गर्न सक्छौं. सो दिस कन्सेप्ट इज भेरी इम्पोर्टेन्ट फ्रम नाउ अन मोर. अहिले हामीले 10 वर्षपछिको समयलाई भिजुअलाइज गर्यौं भने त प्रोबेबली हामीले अहिले जुन रेटमा यो मास डिस्ट्रक्सन भइरहेछ मेडिसिनल प्लान्टको, we must stop this and we should work on this plant. These are some various batches are working together. You some student of some batches, some student of some batches. Balkumari is here. I had a great privilege to have her as a postdoc student. She's going to publish one of the interesting work on orchid. She also contributed. Besides, I have some of the institutions globally, Cells for Science Foundation, Germany, and Marc Selosé, one of the renowned professor from France, Annapurna Research Center, Texas Technical University, some of the international collaborators. Dr. Ranjit is here. We are long collaborator. I have learned a lot from Dr. Ranjit. Thank you so much. Punmaya is here. She has done a marvelous work.
Thank you for your wonderful presentation.
Thank you.
Of course, हाम्रो हिस्ट्रीमा नै मेडिसिनल प्लान्टको एकदमै इम्पोर्टेन्ट छ र मेडिसिनल प्लान्ट्सहरूले दिने जुन सेकेन्डरी मेटाबोलाइट्सहरू छ त्यो चाहिँ हामीले धेरैभन्दा धेरै प्रयोग युज गर्न सक्यौँ भने हाम्रो लागि पनि, हाम्रो देशको लागि पनि र हाम्रो लागि पनि राम्रो हुनेछ होला. यति भन्दै म अब बाँकी रहेको क्वेसन्सहरू हामी लास्टमा क्वेसन अलिअलि एउटा दुईटा क्वेसनहरू हामी त्यही भएर अन्त्यसम्म रहिदिनु होला.
स्टप स्लाइड गरौँ है.
For your wonderful presentation.
Okay. Now we have another presenter, another speaker, Mr. Anmol Mani Mahato. He is a graduate in microbiology and currently working as a executive quality assurance in Bottlers, Nepal. I'd like to request Mr. Anmol Mani to proceed his presentation. Anmol Mani Mahato. Please.
Thank you, sir. Hello, sir. Can you hear me?
Yes,
We are
First of all, good afternoon and namaste. Thanks to the entire team of NRNA Asia Pacific for providing me such a great platform to explore various knowledges, and I feel privileged to take a part in this program. And due to the time limitation, I would like to start. My research topic is isolation and characterization of antibiotic producing actinomycetes isolated from different forest soil of Chitwan district, Nepal. Introduction. Antimicrobial resistance is on the rise globally due to the excessive use of antibiotic. In developing countries such as Nepal, people are using antibiotic even in those minor cases without any medical prescription, due to which antimicrobial resistance is increasing and leads to the treatment failure, and sometimes it may also cause death of the patient. So to overcome from this problem, there is a need in discovery of new antibiotics. And here we have actinomycetes are the bacteria which are potent source of antibiotics, and almost 80% of commercially available antibiotics are isolated from actinomycetes. Objectives of this research is to isolate actinomycetes from different forest soil of Chitwan district, to enumerate the number of actinomycetes isolated, to screen isolated actinomycetes for the production of antibiotics, to determine physiological and biochemical characteristics of antibiotic producing actinomycetes. Here we have methodology. First of all, 1 gram of soil sample was taken, and after that serial dilution was performed. Then split plate was performed on ISP4 media. After incubation, enumeration of actinomycetes were done. And after that, subculture was done on ISP4 media. And then screening of antibiotic producing actinomycetes were done on spot elocution method. And lastly, biochemical power 3, highest number of actinomycetes obtained from Bharatpur Tikoli forest. And 9 different colonies of actinomycetes were obtained from Padampur forest. And out of the 61 different colonies, only 6 were able to show antimicrobial effect and were labeled as sample A, B, C, D, E, and F. Here we can see that antimicrobial effect of isolated actinomycetes against Staphylococcus aureus. Sample A, B, and C has higher inhibition capacity as compared to others. Similarly, antibacterial effect of isolated actinomycetes against Escherichia coli. Here also, sample A, B, and C has higher inhibition capacity. Here in this picture, we can see clear zone of inhibition by sample A towards E. coli. And similarly, we have antibacterial effect of isolated actinomycetes against Salmonella enterica. Here also, sample A, B, and C has higher inhibition capacity as compared to others. In this table, we have antibacterial effect of isolated actinomycetes against Bacillus species. Here, only sample A and B shows higher inhibition capacity towards Bacillus species. And here we have plotted overall inhibition capacity of isolated actinomycetes against the selected bacteria. We can clearly observe that sample A and B shows highly antimicrobial activity against those selected bacteria, whereas sample C shows moderate type of antimicrobial activity, and the sample D, E, and F shows poor antimicrobial activity against those bacteria. Colony color of forward and reverse side was done. Sample A and B has forward side color gray and reverse side color pale yellow, whereas sample C, E, and F has forward side color pale yellow and reverse side color yellow, and sample D has whitish gray forward side color and reverse side color pale yellow. Biochemical test of the actinomycetes. We have performed various biochemical tests such as indole, MR, VB, citrase, urease, motility, catalase, and gelatinase, but mainly focusing on gelatinase production. Sample A and B shows positive result on gelatinase test, whereas sample C shows highly positive result. We can see that in this picture, clear zone of hydrolysis done by sample C. These two are sample A and sample B. Starch hydrolysis and cellulase production test. Sample A, C, and D shows both positive result in starch hydrolysis test as well as cellulase test, whereas other shows negative result. Here in this picture, we can see that clear zone of hydrolysis done by sample D on 1% starch. We also perform a pH tolerance test of the bacteria in various pH range starting from 2 to pH 10. Sample A, C, and D can grow from pH range 2 to pH range 10 as compared to others. Similarly, salt tolerance test of isolated actinomycetes were done from 2% sodium chloride concentration up to 10%. And here only sample E can grow from 2% sodium chloride concentration up to 7.5%. Here, this is the picture of salt tolerance test of isolated actinomycetes. And similarly, we have done growth of actinomycetes on Mekong agar. Except sample D, all other sample were able to grow on Mekong agar. And we also perform antibiotic susceptibility test. Yes, antibiotics such as gentamicin, cloxacillin, ciprofloxacin, ampicillin, and levofloxacin were used. There were no zone of inhibition, only in cloxacillin. This is the picture of antibiotic susceptibility test. Here we can see that no zone of inhibition in cloxacillin. And lastly, I would like to conclude, stain A and B produce very good antibacterial activity against those bacteria which we had already used gram-positive as well as gram-negative bacteria. And antibiotic produced by stain A and B has successfully inhibited both gram-positive as well as gram-negative bacteria. So it was found to be of a broad spectrum type. And during fermentation to produce secondary metabolite, there might be various changes such as fluctuation in pH, temperature, salt concentration, and others. So it will be easy that the isolated actinomycetes can survive in such extreme conditions too. And the last conclusion is that the soil of Chitwan forest consists of abundant number of actinomycetes which can be used in discovery and development of novel antibiotics to combat the threat of antibiotic resistance in the world. Lastly, I would like to thank my supervisor, Mr. Anil Shrestha, lecturer of microbiology, for the guidance and support throughout my research. I would like to express gratitude to Professor Dr. Dheeraj Chaudhary and to the entire team of NRNA Asia Pacific for providing such a great platform. I would also like to thank Head of Department, Mr. Anupuni Bajrasare, my friend who helped me throughout this research, and all the faculty for the cooperation. Thank you.
Obviously, exploring actinomycetes is very valuable in the current state where antibiotic resistance has occurred or like a terror state. Thank you, Anmol Mani, for your nice presentations.
Another presenter is Mr. Aditya Timire. He has completed bachelor's degree in microbiology from Biotechnology College, Nepal. Now, during his bachelor's degree, he was engaged in isolation of trichoderma species and their antibiotic characteristics. Now, I would like to pass this course to Aditya, Mr. Aditya Timire.
Thank you.
Start your presentation.
Thank you, ma'am. It's me, Aditya Gimre, a student of microbiology from Balkumari College. And I'd like to thank NRNA Asia for organizing this conference and providing such a wonderful opportunity. Now, I'd like to begin my presentation without any further delay. The topic of my presentation is isolation of trichoderma from different places of Nepal and observation of their antimicrobial characteristics. Now moving on to the introduction. Since the ancient time, there has been the occurrence of different plant diseases thanks to the fungi like Rhizoctonia solani, Sclerotonia sclerotium, and Alternaria solani, giving rise to different diseases such as collar root, root rot, damping of white mold, and early blight in tomato and potato plant. And the immediate solution used by the farmer at that time was the use of chemical pesticides, which although initially controlled the pathogens, but due to its prolonged use, it became resistant by the pathogens. So there has been the need of alternative solution in which trichoderma plays a vital role as biological control agent, as it is a free-living, opportunistic, avirulent, green spot producing fungi found in soil of all kind and rhizosphere of plant. Rapid in culture, A-express aerial mycelium and production of distinctive conidigenous pustules are its microscopic characteristic, and it also produces different bright conidia and different types of branches. And for the control of the pathogens, it utilizes the several mechanisms such as mycoparasitism, which is a complex process in which it produces the hydrolytic enzyme to hydrolyze the pathogenic cell wall, and it also competes for nutrients with them as it uses its specific iron chelators called CD04s that chelates the iron, and it has been found that the trichoderma that are treated with the salt or in stress condition can show good germination by altering its genetic expression. And thanks to the welding, which first initiated the research activity in trichoderma, leading to increasing in its research activity, and this research wish to find better antibiotic strains to inhibit the disease-causing pathogens. Now I would like to move to us objective. Here we have general objective as to isolate trichoderma from different places of Nepal and observation of their antagonistic property. And under specific objective, we have three different objectives, such as first is the isolation, and second is the antifungal susceptibility test, and third is the antibacterial susceptibility test. Then we have materials and method. In method, we collected different soil from Parsa, Lamjung, Gorkha, Khairani of Nepal during the month of December and January. After the collection of the soil, serial dilution was performed to make the dilution of different concentration. And for the primary isolation, potato dextrose agar along with the antibiotic chloramphenicol and streptomycin sulfate were used. And after the incubation, the fungus showing cultural characteristic of trichoderma were used for secondary isolation. They were also preserved in PDS land, and the viability were tested in every 3 months to check the isolate. And we also observed different morphological characteristics such as color, growth pattern, growth rate, spore formation, and pharid length, width, conidial length, and chlamydospore. And different kind of biochemical tests such as carbohydrate assimilation test, cellulose activity, amylase activity, urease activity, phosphate utilization test was performed to see whether it can survive in the media containing nitrogen, phosphorus, and potassium. Then each of the pure isolate were subjected to fermentation by keeping in PDB broth and in water bath for 14 days. After the fermentation, the isolate were subjected to extraction by following with cotton and filter paper. And after the isolate were obtained, they were mixed with ethyl acetate in separating funnel, and later on, after 1 hour of shaking, the different layers such as organic and aqueous layer were formed, and the stock solution was made using organic layer by adding some methanol. Then antimicrobial activity for fungal and bacterial pathogen were performed by using Muller Hilton agar and potato dextrose potato dextrose agar, and also the white Then after for fungal pathogens, dual culture was performed in which real growth of the pathogen and isolate were observed by keeping them just opposite to each other in the PDA plate. Now moving on to the result. Here you can see the 9 different isolate and their names and their source as well as altitude. For P3A isolate, it was quite difficult to obtain the altitude, so it has been left blank. And here the zone of inhibition of Klebsiella in whale. Here out of the 9 different isolate, only 5 were able to produce zone of inhibition, whereas other isolate were restricted by the pathogens. And among the 5 different isolate inhibiting the pathogen, the highest zone of inhibition were shown by L35 isolate from Longzhong with an inhibition of 23 mm. Now moving on to the zone of inhibition against Staphylococcus aureus in whale, here only 1 isolate from Gorkha was able to show zone of inhibition, whereas other failed to inhibit the pathogen Staphylococcus aureus. And now moving on to the experiment performed in Dix by in the Wartman filter paper. Here out of 9 different isolate, only 3 isolate were able to produce zone of inhibition, with P3T from Persa being the most potent one, and similarly P3A showed an inhibition of 10 mm and P3B2 showed an inhibition of 8 mm. And against the Klebsiella pneumoniae, out of the 9 different isolate, only 5 were able to show zone of inhibition, in which the good inhibition was shown by the isolate from Persa, named P3A, and the isolates such as L35 from Longzhong and GR6W from Gorkha, GR6B from Gorkha, and L36 from Longzhong were unable to inhibit the pathogen. Here you can see the zone of inhibition produced by the pathogen against pathogen pneumonia by P3B1C isolate. And in case of E. coli, none of the isolate were able to inhibit it. Thus, and similar case was in of the Pseudomonas. None of the isolate were able to inhibit it. And now moving on to the dual culture performed against Sclerotinia sclerotiorum, the 9 isolate were able to show inhibition percentage, and the highest was shown by L36, the isolate from Longzhong, with an inhibition percentage of 74. And similarly, two common isolate were shown. Inhibition was shown by P3T with 50% inhibition percentage. Moving on to the activity performed against rhizoctomy. Here also all of the isolates were able to show the inhibition percentage, with 3 isolates showing the common inhibition percentage, such as K32 from NRNA, P3A from Persa, and P3B1C also from Persa, with an inhibition percentage of 44. And on the other hand, the highest was shown by isolate from Gorkha named as GR6B with an inhibition percentage of 72. Here you can see how the fungi, how the isolate overtook the pathogens and was able to inhibit its growth. This is the isolate and these are the pathogens. Similarly, against the pathogen Alternaria solani, all of the isolate were able to show higher inhibition percentage than against any of the pathogens, with the highest shown by P3A and L36 with the inhibition percentage of 89.3 and 86.6, other falling in between them. Here also you can see how the trichoderma isolate inhibit the growth of the fungal pathogens against Alternaria solani. Now moving on to conclusion. This research examined the biological diseases, control ability of trichoderma, especially against several pathogenic fungi and bacteria. Among the isolate, the one that was most effective was Sclerotinia sclerotium with the inhibition of 74.4%, and similarly On the other hand, the isolate GR6W from Gorkha was extremely effective against Rhizoctonia solani and showed an inhibition percentage of 72. Whereas a similar experiment, which include isolate of Nepal, showed 66.08 percentage performed by Bastagoti and his colleagues. Here our isolate was found to be potent than his, and similarly inhibition, similarly highest level of inhibition was shown by the isolate pre-3A with an inhibition of 89 against.
You are short of time. Please conclude your presentation.
This is going to finish, sir. Just 2 minutes. With an inhibition percentage of 89, and common inhibition was shown by pre-3T and GR6W. And when it was compared with the data performed by Sarfaraz Khanal, our isolate were found to be quite potent than obtained by him. Similarly, against the bacteria, the isolate GR6B showed an inhibition diameter of 21 mm, and which was quite potent. And similarly, L35 from Lamjung also showed an inhibition of 23 mm against Klebsiella pneumoniae. Now, moving on to the recommendation, we recommend any further researcher interested in trichoderma research can perform outdoor microbial capacity of trichoderma in field trials. Similarly, more antagonistic character can be observed in more pathogens. Thank you.
Okay. Thank you, Aditya, for your nice presentations. अब हाम्रो अर्को स्पिकर हुनुहुन्छ डाक्टर पुनम Dr. Morrison is a senior scientist in the research-based company named G+ Life Science South Korea. सन् 2013 मा सोल नेशनल युनिभर्सिटीबाट बायोलोजिकल साइन्समा PhD अध्ययन पश्चातमा लामो समयदेखि मोलिकुलर बायोलोजी, प्लान्ट बायोटेक्नोलोजी, क्रिस्पर टेक्नोलोजी, फार्मास्युटिकल प्रोडक्सन र प्लान्ट बेस्ड भ्याक्सिन डेभलपमेन्टमा निरन्तर अनुसन्धानरत हुनुहुन्छ। फ्युचर साइन्टिस्ट अवार्ड 2012 बाट सम्मानित डाक्टर मर्जनका थुप्रै आलेखहरू हामी पढ्न पाउन सक्छौं। विश्व अहिले कोभिड-19 महामारीबाट आक्रान्त छन्। यही आक्रान्तमा वैज्ञानिकहरू भ्याक्सिन डेभलपमेन्टको दौडमा पनि छन्। सँगसँगै यसै सन्दर्भमा डाक्टर मर्जनको पनि अहिले मुख्य अनुसन्धान प्लान्ट बेस्ड भ्याक्सिन फर इन्फ्लुएन्जा एन्ड कोभिड-19 रहेको छ। On behalf of this, I am so glad to welcome Dr. Morrison in this Asia Pacific Knowledge Convention to deliver her speech on the topics vaccine production in plant, a safe and new technology for COVID-19 vaccine. Please, Dr. Morrison, you can proceed.
Okay, धन्यवाद धीरज डाक्टर धीरजलाई। र सबभन्दा पहिले मलाई NRN र यो सेसन 4 को अर्गनाइजेसन कमिटीलाई म धन्यवाद दिन चाहन्छु यो मलाई यो प्लेटफर्म प्रोभाइड गरिदिनु भएकोमा। र आज म यो ठाउँमा यो स्टेजमा यो ठाउँमा मेरा आदरणीय गुरु प्रोफेसर डाक्टर विजय पन्त र टिस्यु कल्चरका पायोनियर डाक्टर रञ्जितसँग एउटै स्टेज शेयर गर्न पाउँदा आफूलाई गर्व महसुस गरिरहेको छु र ढिला नगरिकन म आफ्नो प्रेजेन्टेसन अगाडि बढाउन चाहन्छु। स्लाइड शेयर। ओके, म स्क्रिन शेयर गर्छु। ओके, देख्नु भएको छ मेरो स्लाइड? हेलो,
देखेछ म्याम?
सपनमाया स्लाइड शेयर।
देखेछ।
आज म विशेष गरी बिरुवामा भ्याक्सिन प्रोडक्सन भ्याक्सिन निर्माण गर्ने अथवा उत्पादन गर्ने प्रविधि र त्यो प्रविधिलाई अहिले विश्वमा आक्रान्त बनाएको कोरोना भाइरस विरुद्धको कोभिड-19 भ्याक्सिनलाई एउटा सुरक्षित र एउटा नयाँ प्रविधिको रूपमा प्रयोग गर्ने विषयवस्तुमा मेरो प्रस्तुति केन्द्रित रहनेछ। त्यसैले मेरो आजको प्रस्तुतिमा विशेष गरी भ्याक्सिन भनेको के हो र बिरुवामा हामीले भ्याक्सिन बाहेकका अरु औषधिजन्य चीजहरूलाई कसरी उत्पादन गर्न सकिन्छ र भ्याक्सिन यसरी बिरुवामा उत्पादन गरिएको औषधिजन्य तत्त्वहरू र भ्याक्सिनहरू चाहिँ बजारमा उपलब्ध भइसकेका के छन् र अहिले कोभिडको सन्दर्भमा बिरुवामा बनेका भ्याक्सिनहरूको चाहिँ विकास भइरहेको सन्दर्भ र त्यो भ्याक्सिनहरू क्लिनिकल ट्रायलमा इन्टर गरिसकेको कुरा पनि म प्रस्तुत गर्नेछु र साथसाथै यो चाहिँ बिरुवामा आधारित प्रविधि कतिको चुनौतीपूर्ण छ र कतिको चाहिँ अवसर छ र नेपालको कन्टेक्स्टमा यो हामीले कतिको त्यसलाई अवसरको रूपमा प्रयोग गर्न सक्दछौँ भन्ने कुरालाई म प्रस्तुत गर्नेछु। ओके, भ्याक्सिन हामीले कोरोनाको महामारी फैलिएसँगै भ्याक्सिनको प्रचार पनि धेरै भएको छ र साधारण मानिसहरूमा पनि भ्याक्सिनको नलेज चाहिँ अपग्रेड भएकोमा त्यो एउटा हामीले सबल पक्षको रूपमा लिनुपर्दछ र सामान्यतया हामीले भ्याक्सिन भनेको कुनै पनि त्यस्तो चिज हो जुन चाहिँ हाम्रो शरीरमा प्रवेश गरिसकेपछि या चित्रमा देखाएको छ शरीरमा प्रवेश गरिसकेपछि त्यो चिजले हाम्रो शरीरको इम्युन सिस्टमलाई स्टिमुलेट गर्दछ र त्यो कुनै पनि रोग विरुद्धलाई लड्नको लागि एन्टिबडी उत्पादन गर्न सक्दछ भने हामीले त्यस्तो चिजलाई चाहिँ एन्टिबडी हामी चाहिँ भ्याक्सिन भन्न सक्दछौँ र त्यसले गर्दाखेरि भ्याक्सिन भनेको त्यो कुनै पनि रोग लगाउने चिजको कारक तत्त्वको पूर्ण अंश अथवा चाहिँ आंशिक अंशबाट मात्रै बन्न सक्दछ र त्यसरी बन्ने भएकोले गर्दाखेरि त्यो भ्याक्सिन लगाइसकेको मान्छेलाई पछि प्राकृतिक रूपमा त्यो ब्याक्टेरिया वा भाइरसले आक्रमण गर्दाखेरि पहिल्यै चाहिँ निर्माण भइसकेको एन्टिबडीले पनि काम गर्दछ र त्यो एन्टिबडी बन्ने क्रममा जुन चाहिँ हाम्रो इम्युन सिस्टमले मेमोरीको रूपमा राखेको हुन्छ त्यो मेमोरीले गर्दाखेरि पनि नेचुरल चाहिँ भाइरसहरू इन्टर गर्दाखेरि अझ द्रुत गतिमा छिटोभन्दा छिटो चाहिँ एन्टिबडी प्रोडक्सन गरेर भ्याक्सिन लगाइसकेको मानिसलाई चाहिँ रोग विरुद्ध चाहिँ प्रोटेक्सन दिने काम गर्दछ। त्यसलाई नै हामी चाहिँ भ्याक्सिन अथवा भ्याक्सिनेसन भन्दछौँ। यसरी हेर्दाखेरि संसारमा अहिले चाहिँ धेरै भ्याक्सिनहरूको डेभलप भइसकेको छ र त्यस्ता विकास भएका भ्याक्सिनहरूले गर्दाखेरि मानव जातिलाई 25 भन्दा पनि बढी रोगहरूबाट चाहिँ बचाएको छ र यसमा अहिले लिस्टमा अब कोरोना भाइरस विरुद्धको भ्याक्सिन छिटै आउँदैछ भन्ने हामीले आशा गर्न सक्दछौँ। र यसरी चाहिँ अहिले उपलब्ध भ्याक्सिनहरू र विकास हुने क्रममा रहेका भ्याक्सिनहरूलाई हामीले चाहिँ विभिन्न वर्गीकरण गर्न सक्दछौँ विभिन्न प्रकारमा, जस अन्तर्गत कन्भेन्सनल भ्याक्सिनमा होल इनएक्टिभेटेड भ्याक्सिन जसमा चाहिँ रोग लगाउने कारक तत्त्वको पुरै चिज प्रयोग गरिएको हुन्छ र त्यसलाई चाहिँ निष्क्रिय रूपमा प्रयोग गरिएको हुन्छ अथवा मृत रूपमा प्रयोग गरिएको हुन्छ भने त्यसपछि विकास भएको भ्याक्सिनलाई हामी चाहिँ सब युनिट भ्याक्सिन रिकम्बिनेन्ट भ्याक्सिन भन्छ जसमा त्यो कारक तत्त्वको पुरै अंश प्रयोग नगरिकन केही अंश जुनले एन्टिजेनको काम गर्न सक्दछ त्यो चिजलाई मात्र प्रयोग गरिएको भ्याक्सिनलाई हामी रिकम्बिनेन्ट सब युनिट भ्याक्सिन भनिन्छ। र अहिले न्यू जेनेरेसन भ्याक्सिन अन्तर्गत थुप्रै भ्याक्सिनहरूको चाहिँ विकास भइरहेको छ। त्यस अन्तर्गत रिकम्बिनेन्ट भाइरल भेक्टर भनेको अहिले हामीले सुन्ने गरेका छौँ चाइनाबाट उत्पादन भएको कोरोना विरुद्धको भ्याक्सिन चाहिँ एडी5 भ्याक्सिन भनेर एडिनोभाइरस 5 भ्याक्सिन भनेर सुन्ने गरेका छौँ। त्यो भनेको एडिनोसिन भ्याक्सिन भाइरसमा सार्सको जेनेटिक म्याटेरियललाई इन्ट्रोड्युस गराएर त्यो भाइरसलाई चाहिँ भेक्टरको रूपमा प्रयोग गरेर बनाइएको भ्याक्सिनलाई चाहिँ त्यसरी रिकम्बिनेन्ट भाइरल भेक्टर भ्याक्सिन भनिन्छ। र त्यो बाहेक पनि ब्याक्टेरियालाई पनि भेक्टरको रूपमा प्रयोग गरेर त्यसरी भ्याक्सिन बनाउन सकिन्छ। र अर्को डिएनए भ्याक्सिन भनेको एन्टिजेनको रूपमा प्रयोग गर्ने प्रोटिनलाई त्यो प्रोटिन नै नभइकन जस्ट त्यो प्रोटिन बन्न चाहिने डिएनएलाई प्रयोग गरेर पनि भ्याक्सिन बनाइन्छ। र अहिले मोडेनाको जुन चाहिँ क्लिनिकल ट्रायल 3 मा पुगेको छ त्यो भ्याक्सिन मोडेना कम्पनीले बनाएको भ्याक्सिन चाहिँ यो डिएनए भ्याक्सिन अन्तर्गत पर्दछ। यसरी हेर्दाखेरि रिकम्बिनेन्ट भ्याक्सिन टेक्नोलोजी चाहिँ धेरै वर्ष अघिदेखि विकास भएको। रिकम्बिनेन्ट डिएनए भ्याक्सिन भन्नाले रिकम्बिनेन्ट डिएनए टेक्नोलोजी प्रयोग गरेर भाइरस या ब्याक्टेरियामा अथवा अरू कारक तत्त्वहरूको जेनेटिक म्याटेरियललाई लिएर त्यो एन्टिजेन प्रड्युस गरेर त्यसबाट बनाइएको भ्याक्सिनलाई हामी रिकम्बिनेन्ट भ्याक्सिन भन्न सक्दछौँ। र त्यसरी त्यो डिएनएबाट प्रोटिन बनाउनको लागि ब्याक्टेरिया अथवा इन्सेक्ट अथवा यीस्ट म्यामेलियन सेल वा प्लान्ट सेललाई चाहिँ त्यो प्रोटिन म्यानुफ्याक्चरिङ फ्याक्ट्रीको रूपमा चाहिँ प्रयोग गरिन्छ। यसरी 1986 मा नै रिकम्बिनेन्ट भ्याक्सिन चाहिँ ह्युमन बिङको युजको लागि चाहिँ एप्रुभ भएर अहिले धेरै प्रकारको रिकम्बिनेन्ट भ्याक्सिनहरू हामीले मार्केटमा पाउन सक्दछौँ। र अब यसरी चाहिँ भ्याक्सिनहरू बनाउने विभिन्न प्रविधिहरू छन्। त्यसमध्येमा सबैभन्दा परम्परागत भ्याक्सिनको रूपमा हामीले यो एग बेस्ड टेक्नोलोजीलाई लिन सक्दछौँ। यो 70 वर्षभन्दा पुरानो टेक्नोलोजी हो र यसमा चाहिँ त्यो कारक तत्त्व विशेष गरी भाइरस बनाउनु, उदाहरणको लागि भाइरसलाई चाहिँ फर्टिलाइज एगभित्र त्यसलाई ग्रो गराइन्छ, मल्टिप्लाई गराइन्छ र त्यहाँबाट निकालिसकेपछि कि एटेन्युएटेड गरिन्छ या चाहिँ त्यसलाई इनएक्टिभेटेड गरेर भ्याक्सिनको रूपमा फर्मुलेसन गरिन्छ। र यसको सबल पक्ष र दुर्बल पक्ष भनेको चाहिँ यो निकै सस्तोमा बनाउन सकिन्छ, तर यसको लागि धेरै लामो समय, कम्तीमा पनि 6 महिना लाग्ने हुन्छ। र अर्को यसरी ग्रो गराउँदाखेरि मानिसलाई चाहिँ रोग लगाउन सक्ने प्याथोजेनहरूको कन्टामिनेसन हुने चान्स हुन्छ र अन्डाभित्र ग्रो हुँदाखेरि त्यो भाइरस या ब्याक्टेरियामा म्युटेसन भएर त्यसले नकारात्मक असर पुर्याउने सम्भावना पनि हुन्छ र अन्डाबाट एलर्जी हुने मान्छेहरूलाई यसरी एक टेक्नोलोजीबाट बनाएको भ्याक्सिन चाहिँ एलर्जिक हुने गर्दछ। र त्यसपछि विकास भएको टेक्नोलोजी भनेको सेल बेस्ड टेक्नोलोजी हो। सेल बेस्ड टेक्नोलोजीमा अन्डाको ठाउँमा चाहिँ हामी सेलभित्र चाहिँ त्यो भाइरस या ब्याक्टेरियालाई ग्रो गराइन्छ र जुन अन्डाको कम्पेयरमा चाहिँ धेरै फास्ट हुन्छ, विदिन फ्यु विक्समा चाहिँ त्यो भ्याक्सिन प्रड्युस गर्न सक्ने हुन्छौँ। तर यसको अर्को पक्ष भनेको चाहिँ त्यो निकै महँगो पर्दछ। जुन चाहिँ सेल ग्रो गर्ने सिस्टमहरू छ त्यो निकै महँगो पर्दछ। त्यसले गर्दाखेरि यो चाहिँ राम्रो भए तापनि पैसाको हिसाबले अथवा खर्चको हिसाबले चाहिँ यसको चाहिँ अर्को दुर्बल पक्ष मान्न सकिन्छ। त्यसपछि अब यसको अल्टरनेटिभमा के हो त भन्दाखेरि त्यसको अल्टरनेटिभमा मानिसले विकास गरेको अर्को प्रविधि भनेको प्लान्ट बेस्ड रिकम्बिनेन्ट भ्याक्सिन टेक्नोलोजी हो। ल प्लान्टलाई हामीले भ्याक्सिन मात्र नभएर अन्य कुनै पनि औषधिहरू प्रयोग गर्नको लागि फ्याक्ट्रीको रूपमा प्रयोग गर्न सक्दछौँ। जस्तै एन्टिबडी प्रड्युस गर्न सक्छौँ हामीले, त्यसपछि चाहिँ इन्जाइमहरू प्रयोग गर्न उत्पादन गर्न सक्छौँ र अरू चिजहरू जुन चाहिँ हामीले अरू सिस्टममा जस्तै एग सिस्टममा अथवा चाहिँ इकोलाइज सिस्टममा चाहिँ प्रड्युस गर्न सक्दैनौँ त्यस्ता खालको कम्प्लेक्स टाइपको प्रोटिनहरूलाई पनि हामीले चाहिँ प्लान्टमा चाहिँ प्रड्युस गर्न सक्दछौँ। र अर्को यो प्लान्ट बेस्ड प्रविधिको सबभन्दा सबल पक्ष भनेको इडिबल भ्याक्सिन हो। इडिबल भ्याक्सिन जस्तै हामीले चाहिँ खान योग्य बिरुवालाई प्रयोग गरेर भ्याक्सिन बनायो भने त्यो बिरुवालाई नै हामीले चाहिँ भ्याक्सिनको रूपमा कन्ज्युम गर्न सक्दछौँ, जसलाई इडिबल भ्याक्सिनको रूपमा हामी प्रयोग गर्न सक्छौँ। जसले गर्दा हामीलाई अहिले भ्याक्सिन लगाउनलाई भ्याक्सिन चाहिँ प्रिजर्भेसनको लागि जुन चाहिँ कोल्ड चेन सिस्टमको आवश्यकता पर्दछ त्यो आवश्यकता हुँदैन। त्यसले गर्दाखेरि दूर दराजमा रहेका पपुलेसनलाई पनि हामीले सजिलैसँग इम्युनाइजेसन गर्न सक्षम हुनेछौँ। प्लान्ट बेस्ड फार्मास्युटिकलको हिस्ट्री धेरै लामो भइसकेको छैन, लगभग 40 वर्ष मात्रै भएको छ र त्यो 40 वर्षको भित्रमा पनि प्लान्ट बेस्डमा काम गरेका साइन्टिस्टहरूको ठुलो लगावले गर्दाखेरि प्रगति धेरै भइसकेको हामीले मान्न सक्दछौँ। 2006 सालमा नै भेटनरी पर्पोजको लागि प्लान्टमा बनेको भ्याक्सिन अप्रुभ भएर मार्केटमा आएको थियो र ह्युमन पर्पोजको लागि बनेको औषधि भने 2012बाट मार्केटमा आइसकेको छ। यसरी अरू औषधिहरूको सन्दर्भमा पनि त्यत्तिकै नै आवश्यकता छ र त्यस्तै नै महत्त्व छ, उपयोगी छ प्लान्ट बेस्ड टेक्नोलोजी। र भ्याक्सिनको लागि अझ बढी किन आवश्यक छ भन्दाखेरि अघि नै भने जस्तै भ्याक्सिन बनाउन एक बेस्ड प्रविधि प्रयोग गर्दाखेरि निकै लामो समय लाग्दछ। जस्तै त्यो एन्टिजेन प्रड्युस गर्ने, भाइरस प्रड्युस गर्न लगभग 75 दिन जति लाग्दछ। तर त्यही प्रविधिलाई हामीले चाहिँ एन्टिजेन प्रड्युस गर्न प्लान्ट बेस्ड सिस्टम युज गर्यो भने 21 दिन मात्रै हुन्छ। त्यो भनेको 3 गुणाले फास्टर हामीले चाहिँ प्लान्टमा भ्याक्सिन तयार गर्न सक्दछौँ। र कस्टको हिसाबमा हेर्ने हो भने यो अघि सेल बेस्ड टेक्नोलोजी भन्दा 220 गुणाले सस्तो पर्दछ प्लान्टमा भ्याक्सिन बनाउँदा भने एक बेसको भन्दा 10 गुणाले चाहिँ सस्तो पर्न जान्दछ। त्यसले गर्दाखेरि हामी जस्तो भनौँ न डेभलप हुँदै गरेको कन्ट्रीहरूको लागि अझ यो प्रविधिको महत्त्व अझ बढी छ। अब प्लान्ट यसरी चाहिँ यति चाहिँ उपयोगी, यति सस्तोमा प्रड्युस हुने भ्याक्सिन बनाउने प्रक्रियाहरूको बारेमा म सामान्यतया चाहिँ तपाईलाई जानकारी गराउन चाहन्छु। र प्लान्ट बेस्ड भ्याक्सिन प्रोडक्सनको बेस भनेको फाउन्डेसन भनेको चाहिँ हाम्रो रिकम्बिनेन्ट डिएनए टेक्नोलोजी हो। र यो टोटल प्रोसेसलाई हामीले 3 भागमा डिभाइड गर्न सक्छौँ, अपस्ट्रिम प्रोसेस, प्रोटिन प्रोडक्सन प्रोसेस र डाउनस्ट्रिम प्रोसेस। त्यसमा अपस्ट्रिम प्रोसेसमा हामीले चाहिँ जुन एन्टिजेन बनाउनु पर्ने हो त्यो एन्टिजेनको लागि रिस्पोन्सिबल जिनलाई चाहिँ हामीले लिएर क्लोनिङ गर्छौँ र त्यसलाई चाहिँ प्लान्टको सेलमा इन्ट्रोड्युस गराउँछौँ र प्लान्टको सेलमा इन्ट्रोड्युस गराइसकेपछि प्लान्टभित्रको जुन प्रोटिनको चाहिँ मेसिनरी छ, प्रोटिन बनाउने मेसिनरी युज गरेर त्यसले चाहिँ प्रोटिन बनाउँदछ र यदि हामीले यो प्लान्टको होस्ट प्लान्टको रूपमा इडिबल प्लान्ट युज गर्यो भने सिधै त्यसलाई इडिबलको भ्याक्सिनको रूपमा प्रयोग गर्न सक्छौँ। जस्तै चाहिँ अब यो सागलाई, आलु, मकै र चाहिँ टमाटोलाई चाहिँ होस्ट प्लान्टको युज गर्यो भने इडिबल भ्याक्सिनको प्रयोग गर्न सक्दछौँ। यदि सुर्ती जस्ता प्लान्ट जुन चाहिँ हामी कन्ज्युम गर्न सक्दैनौँ, नन इडिबल छ, त्यसको केसमा हो भने त्यसलाई हामी एक्सट्र्याक्ट गर्छौँ र त्यसपछि प्रशोधन प्युरिफिकेसन गरिसकेपछि त्यसलाई भ्याक्सिनको रूपमा चाहिँ हामीले प्रयोग गर्न सक्दछौँ। र त्यसरी गर्दाखेरि चाहिँ सबभन्दा अर्को महत्त्वपूर्ण चाहिँ स्टेप भनेको रिकम्बिनेन्ट भ्याक्सिन बनाउँदाखेरिको महत्त्वपूर्ण स्टेप भनेको चाहिँ जुन चाहिँ त्यो बाह्य चिजबाट आएको डिएनए छ त्यो डिएनएलाई बिरुवाको सेलसम्म इन्ट्रोड्युस गराउनु अर्को एउटा महत्त्वपूर्ण स्टेप हो र त्यो गर्नको लागि हामी ब्याक्टेरियाको एउटा जाति अग्रोब्याक्टेरियम भन्ने प्रयोग गर्दछौँ र अग्रोब्याक्टेरियमलाई चाहिँ बिरुवाको सेलसम्म पुर्याएर त्यसमा चाहिँ एन्टिजेन प्रोडक्सन गर्ने दुईवटा मेथडहरूमा हामी डिभाइड गर्न सक्छौँ। एउटा स्टेबल ट्रान्सफर्मेसन, अर्को ट्रान्जिएन्ट ट्रान्सफर्मेसन। स्टेबल ट्रान्सफर्मेसनमा चाहिँ हामीले यसरी चाहिँ बाह्य जुन डिएनए हुन्छ त्यसलाई लिएर बिरुवाको जुन चाहिँ जेनोम हुन्छ, टोटल जिनहरूको सिक्वेन्स, त्यसमा चाहिँ जेनोममा इन्टिग्रेसन गर्छौँ हामी, पर्मानेन्टली इन्टिग्रेसन गर्दछौँ। त्यसले गर्दाखेरि हामी चाहिँ त्यसबाट ट्रान्सजेनिक प्लान्ट बनाउँछौँ र त्यसबाट सिड लिइसकेपछि हामी जेनेरेसन टु जेनेरेसन चाहिँ त्यो भ्याक्सिन अथवा चाहिँ अरू औषधिहरू पाइराख्छौँ। र अर्को मेथड भनेको त्यो प्लान्टबाट हामी सिड यसरी सेल लाइन बनाएर सेल कल्चर गरेर हामीले धेरै समयसम्म चाहिँ बिरुवा पाएकै त्यो एन्टिजेन प्रोडक्सन गर्न सक्छौँ। र अर्को मेथड भनेको जस्तै यस्तो यो प्रविधिबाट हामीले एन्टिजेन वा औषधिजन्य पदार्थ पाउनको लागि चाहिँ धेरै लामो समय लाग्छ, कम्तीमा 2 वर्ष लाग्न सक्दछ र अहिलेको जस्तो कोभिडको समयमा यो प्रविधि त्यति उपयोगी नहुन सक्दछ। कोभिड जस्तो केसमा चाहिँ पेन्डेमिकको समयमा तुरुन्त हामीलाई भ्याक्सिन चाहिने भएकोले त्यसको लागि ट्रान्जिएन्ट ट्रान्सफर्मेसनले काम गर्दछ। यसमा चाहिँ टेम्पोरेरी हामीले चाहिँ डिएनएलाई इन्ट्रोड्युस गराएर प्रोटिन चाहिँ विदिन 1 to 2 विक्समा चाहिँ हामी प्रोटिन प्रड्युस गराउन सक्छौँ र त्यो प्युरिफिकेसन गरेर त्यसलाई हामीले चाहिँ अप्लाई गर्न हामीले एप्लिकेसनमा लान सक्दछौँ। अब यसरी प्लान्ट बेसमा भएर प्रयोग गरिएको उत्पादन गरिएको चाहिँ फार्मास्युटिकल्सहरू चाहिँ मार्केटमा आइसकेको छ अहिले। जस्तै धेरै चिजहरू चाहिँ इन्डस्ट्रियलको रूपमा इन्डस्ट्रियल इन्जाइमहरू, प्रोटिनहरू धेरै आइसकेको छ र त्यसमा ह्युमन डिजिजको लागि चाहिँ किन भन्दाखेरि ह्युमन डिजिज कन्ट्रोलको लागि आउने चिजहरूमा चाहिँ धेरै कन्ट्रोल हुन्छ, सेफ्टी म्याटरहरू हुने भएकोले गर्दाखेरि 2012मा फर्स्ट टाइम चाहिँ यो प्लान्टमा बनेको मेडिसिनले चाहिँ एप्रुभ भएको र एप्रुभ भएर अहिले मार्केटमा आएको छ। त्यो चाहिँ ट्यालिग्लुसेरेट अल्फा हो। यो गौचर डिजिजको ट्रिटमेन्टको लागि प्रयोग गरिन्छ र यसलाई प्रोटालिक्स कम्पनीले चाहिँ डेभलप गरेर अहिले फाइजरले चाहिँ मार्केटिङ गरेको छ। र यो कसरी बनायो भन्दाखेरि हामीले सधैँ खाने गाजर छ, त्यो गाजरको सेलमा उत्पादन गरिएको औषधि हो। यो गाजरको सेलबाट लिफलाई प्रयोग गरेर यसरी चाहिँ अघि नै म्याडमले पनि देखाइसक्नु भएको छ, टिस्यु कल्चरको एउटा प्रोसेस हो। यो टिस्यु कल्चर गरिन्छ र यसरी डेभलप भएको सेलहरूमा चाहिँ अग्रोब्याक्टेरियममा रहेको जुन चाहिँ त्यो इन्जाइम प्रड्युस गर्न चाहिने जिन हो त्यो जिनलाई यहाँ इन्ट्रोड्युस गराइन्छ र सेल लाइन प्रड्युस गरिन्छ र यो सेलहरूलाई यस्तो ठुल्ठुलो ट्याङ्कहरूमा सेल कल्चरको यो कल्चर ट्याङ्कहरूमा कल्चर गरेर त्यहाँबाट एक्सट्र्याक्ट गरेर चाहिँ अहिले यो औषधिको रूपमा बजारमा उपलब्ध छ। त्यस्तै गरी अर्को एउटा उदाहरण प्रस्तुत गर्न चाहन्छु। 2014 सालमा इबोलाले हजारौं मानिसहरूको मृत्यु भएको थियो। त्यति बेला इबोलाको अहिले कोरोनाको जस्तै कुनै पनि औषधिहरू थिएन र त्यतिखेर रिसर्चकै कन्डिसनमा रहेको चाहिँ यो प्लान्ट बेस एन्टिबडी औषधि थियो। औषधि चाहिँ रिसर्च हुँदै थियो र त्यो चाहिँ एकदमै इमर्जेन्सी केस भएकोले गर्दाखेरि क्लिनिकल ट्रायल विदाउट क्लिनिकल ट्रायल नै चाहिँ त्यो चाहिँ फास्ट ट्र्याक क्लिनिकल ट्रायलको रूपमा लिएर मानिसहरूमा 7 जना मानिसहरूमा त्यो औषधि प्रयोग गरिएको थियो र 7 जनालाई नै त्यसले चाहिँ बचाउन चाहिँ सहयोग गरेको थियो। तर दुर्भाग्य भन्नुपर्दछ यो रिसर्च स्टेजमा रहेको यो औषधि चाहिँ 7 जनालाई मात्र पर्याप्त भयो, त्यो मात्रा नपुगेर अरूले उपचार पाउन सकेन। र यो कसरी बनाएको थियो भन्दाखेरि यो चाहिँ सुर्तीको बिरुवामा जुन चाहिँ यो औषधि एन्टिबडी प्रयोग गर्न एन्टिबडी प्रड्युस गर्ने जिनलाई इन्ट्रोड्युस गराएर यो 3 वटा एन्टिबडीहरूको कम्बिनेसनबाट बनेको औषधि हो जिम्याप। त्यसको लागि 3 वटा जिनहरू प्रोटिनहरू इन्ट्रोड्युस गराएर त्यहाँबाट चाहिँ सुर्तीको बिरुवाबाट उत्पादन गरिएको हो। त्यसलाई चाहिँ अघि जस्तै पेन्डेमिक भएकोले गर्दा ट्रान्सजेनिक होइन, पर्मानेन्ट होइन, टेम्पोररी ट्रान्जिएन्ट एक्सप्रेसन सिस्टम प्रयोग गरेर त्यसलाई चाहिँ उत्पादन गराएको थियो। यसरी
डाक्टर मर्जन, वी आर शॉर्ट अफ टाइम प्लिज।
ओके, सो यसरी हेर्दा हामीले लिस्टमा हेर्ने हो भने धेरै प्लान्ट, धेरै चिजहरू चाहिँ अहिले धेरै प्रडक्टहरू चाहिँ क्लिनिकल फेजको डिफ्रेन्ट स्टेजमा छ। त्यसमा एउटा भन्न चाहन्छु, अहिले रिसेन्टली जुलाईमा चाहिँ यो इन्फ्लुएन्जा विरुद्धको भ्याक्सिन चाहिँ क्लिनिकल ट्रायल 3rd स्टेज चाहिँ कम्प्लिट भइसकेको छ। त्यसको अर्थ हामी छिटै चाहिँ मार्केटमा पाउँदैछौँ। र अब कोरोनाको विषयलाई हेर्दाखेरि कोरोनामा चाहिँ सबै संसारका मान्छेहरू भ्याक्सिन बनाउन दौडिरहेको बेलामा प्लान्ट बेस्ड भ्याक्सिनमा काम गर्ने साइन्टिस्टहरू पनि त्यतिकै मेहनत गरिरहेका छन्। र अहिलेको अवस्थामा जी+ लाइफ साइन्सेस जुन चाहिँ म आफू काम गर्दछु त्यसले हामीले पनि भ्याक्सिन बनाउन प्रयास गरेका छौँ र अहिले हाम्रो प्रडक्टले चाहिँ प्री क्लिनिकल फेज चाहिँ कम्प्लिट गरिसकेको छ। र त्यस्तै गरी क्यानाडाको मेडिकागो र युएसएको केन्टुकीले चाहिँ क्लिनिकल फेज फर्स्टमा चाहिँ इन्टर गरिसकेको छ। र हाम्रो सिस्टम, हाम्रो कम्पनीको सिस्टम भनेको चाहिँ त्यसको विशेषता भनेको चाहिँ ह्युमनाइज होस्ट प्लान्ट हो। त्यो भनेको चाहिँ ह्युमनको र प्लान्टको चाहिँ त्यो ग्लाइकोजाइलेसन प्याटर्न डिफ्रेन्ट हुन्छ। त्यसले गर्दा प्लान्टमा प्रड्युस भएको प्रोटिनहरूले कहिलेकाहीँ ह्युमनहरूमा चाहिँ एलर्जिक प्रड्युस गर्छ। त्यसले गर्दा हामीले चाहिँ त्यस्तो एलर्जी नहोस् भनेर बिरुवालाई ग्लाइको इन्जिनियर गरेर क्रिस्पर-कास सिस्टम प्रयोग गरेर त्यसको ग्लाइकोजाइलेसन चाहिँ इन्जिनियर गरेका छौँ। त्यसले गर्दा ह्युमनको जस्तै ग्लाइकोजाइलेसन प्याटर्न तयार भएको एन्टिबडी हामी प्रड्युस गर्दछौँ। र यसरी चाहिँ हामी मास स्केलमा चाहिँ प्लान्टहरू प्रड्युस गर्छौँ। लगभग हामी एउटा लटमा नै लगभग 12,000 प्लान्टहरू प्रड्युस गर्छौँ। र त्यसरी लार्ज स्केलमा हामी एक्सट्र्याक्सन गरेर र प्युरिफिकेसन आफ्टर चाहिँ यसरी चाहिँ हामी प्री क्लिनिकल ट्रायलमा चाहिँ इन्ट्री गरिसकेका छौँ। र अर्को चाहिँ यो VLP भाइरस लाइक पार्टिकल्स भनेको चाहिँ मेडिकागोको चाहिँ यो प्लेटफर्म हो। यसमा चाहिँ यो अझ बढी इफेक्टिभ हुन्छ। यसमा चाहिँ के हुन्छ भन्दा भाइरस जस्तै पार्टिकल बनाउँछ। यसमा तपाईँले चित्रमा देख्न सक्नुहुन्छ। यो चाहिँ साँच्चीकै भाइरसको, कोरोना भाइरसको चाहिँ चित्र हो भने यो चाहिँ मेडिकागो कम्पनीले सर्टेन चाहिँ प्रोटिनहरू युज गरेर बनाएको भाइरस लाइक पार्टिकल हो। यो अझ बढी इफेक्टिभ हुन्छ र यसले चाहिँ अझ लङ लास्टिङ इम्युन सिस्टम दिन्छ। र यो प्रडक्ट अहिले चाहिँ क्लिनिकल 1मा चाहिँ इन्टर गरिसकेको छ। र यसलाई बनाउने सिस्टम भनेको प्रोडक्सन सिस्टम चाहिँ अघि नै भनेको जस्तै इन्ट्रेस्ट एजेन्ट इन्ट्रोड्युस गराउँछ प्लान्टमा र ट्रान्जिएन्ट सिस्टम भनौँ न टेम्पोररी प्रड्युसन गरेर त्यसलाई चाहिँ टेम्पोररी सिस्टम प्रयोग गरेर प्रोडक्सन गर्छ र प्युरिफिकेसन गर्छ। र त्यसबाहेक पनि यो कम्पनीले अरू प्रडक्टहरू पनि उसको विभिन्न क्लिनिकल ट्रायलमा पुगिसकेको छ, 2nd, 1st यसरी। र यसको च्यालेन्जेसहरू अब चाहिँ के छ भन्दाखेरि जस्तै अब अहिलेसम्म भ्याक्सिन त आएको छैन, अरू धेरै औषधि आइसकेको भए पनि र यसको चाहिँ एउटा च्यालेन्ज भनेको लो यिल्ड छ। प्लान्टमा चाहिँ हेटेरोजिनस प्रोटिन भनौँ, अर्को सोर्सबाट आएको डिएनएलाई प्रोटिनमा ट्रान्सलेसन गर्ने पोटेन्सियल चाहिँ अलिक कम भएकोले त्यसमा चाहिँ च्यालेन्जिङ छ। अनि प्युरिफिकेसन मेथडहरू चाहिँ इकोलाइमा जस्तो त्यति इस्टाब्लिस नभइसकेकोले गर्दाखेरि अझै चाहिँ हामीले चाहिँ मोडिफिकेसन गर्दै त्यसलाई चाहिँ हिट एन्ड ट्रायलमाबाट चाहिँ अलिक चाहिँ अझै चाहिँ स्टेबल सिस्टम इस्टाब्लिस गर्न अझै पनि टाइम बाँकी छ। अर्को अवसर भनेको चाहिँ के भन्दाखेरि अहिले नै हामीले चाहिँ अघि नै भने जस्तै चाहिँ औषधिहरू आइसकेको छ बजारमा र धेरै प्रडक्टहरू क्लिनिकल ट्रायलमा गइसकेको छ। त्यसले गर्दा हामी आशावादी बन्न सक्छौँ। र नेपालको सन्दर्भमा अघि जस्तै ट्रान्सजेनिक औषधि बनाउने ट्रान्सजेनिक लाइन बनाएर एग्रिकल्चरलाई ट्रेडिसनल एग्रिकल्चरको ठाउँमा हामी मेडिसिनल एग्रिकल्चरलाई युज गरेर इकोनोमिक डेभलपमेन्टमा अगाडि बढ्न सक्दछौँ। र लोकल प्रडक्सनहरू गरेर चाहिँ हामीले चाहिँ दूरदराजमा रहेका मानिसहरूलाई चाहिँ यो औषधिहरू उपलब्ध चाहिँ सस्तो रूपमा औषधिहरू उत्पादन गर्न सक्नेछौँ। र सबभन्दा महत्त्वपूर्ण कुरा भनेको चाहिँ जुन चाहिँ अहिले चाहिँ कोल्ड चेन सिस्टम प्रयोग गरेर भ्याक्सिनहरू बनाउनु पर्ने छ, त्यसले गर्दाखेरि फ्रिज नभएको ठाउँमा हामीले चाहिँ भ्याक्सिनलाई बचाउने जुन समस्या छ त्यो समस्याबाट हामीले चाहिँ इडिबल भ्याक्सिन प्रयोग गरेर त्यो हटाउन सक्दछौँ। र नेपालको कन्टेक्स्टमा चाहिँ अब के गर्न सक्दछौँ भन्दाखेरि मैले 3 भागमा बाँडेको छु यहाँ। जस्तै प्लान्ट एक्सप्रेसन भेक्टर सिस्टम प्रयोग गर्न सक्छौँ। यो भनेको मैले भने अघि नै बिरुवामा चाहिँ अन्तबाट जस्तै भाइरसबाट आएको जिनलाई प्रोटिनमा डेभलप गर्नलाई एकदमै गाह्रो छ र प्रोडक्सन लेभल एकदमै लो हुन्छ। र त्यो गर्ने भनेको चाहिँ त्यो जुन डिएनएलाई चाहिँ बिरुवामा लग्ने जुन भेक्टर छ त्यो भेक्टर इज द मोस्ट इम्पोर्टेन्ट हुन्छ। र त्यो भेक्टर डेभलप गर्नलाई हामीले चाहिँ धेरै रिसर्च गर्नु पर्दैन। त्यो चाहिँ डेटाबेस अनि त्यसपछि चाहिँ हाम्रो चाहिँ थ्योरिटिकल नलेजहरू चाहिँ धेरै चाहिन्छ। त्यसको लागि नेपालमा म धेरै सम्भावना देख्दछु। जुन भनौँ न मानिसहरू चाहिँ आइडिया भएका मान्छेहरू धेरै स्टडी गर्न सक्ने मान्छेहरूले लिटरेचरको आधारमा चाहिँ कसरी प्रोटिनलाई प्रड्युस बढी गराउन सकिन्छ, एमआरएनए लेभल कसरी बढाउन सकिन्छमा धेरै स्टडी गरेर हामीले बनाउन सक्दछौँ। यो अलिकति च्यालेन्जिङ छ, तर यो धेरै चाहिँ इकोनोमिकल भ्यालु छ। जस्तै एउटा चाहिँ अहिले जापानको डेङ्का भन्ने कम्पनी छ, औषधि बनाउने कम्पनीले आइकोन जेनेटिक्स भन्ने एउटा सानो कम्पनीले विकास गरेको भेक्टरलाई चाहिँ उसले एउटा भेक्टरको लागि उसले $75 मिलियन तिरेर किनेको छ 2017 मा। त्यसले गर्दाखेरि मलाई लाग्छ नेपालका विद्यार्थी भनौँ अहिले चाहिँ ग्रेजुएट स्टुडेन्टलाई यो एउटा ठुलो चाहिँ अवसर हुन सक्दछ। र अर्को भनेको चाहिँ डेभलपमेन्ट अफ प्लान्ट इन्क्लुडिङ क्रप विथ द हाई भ्यालु ट्रेड्स। जस्तै हामीले त्यो जुन रिकम्बिनेन्ट डिएनए टेक्नोलोजी प्रयोग गरेर र प्रोटिन एक्सप्रेसन सिस्टम प्रयोग गरेर हामीले चाहिँ स्टेबल एक्सप्रेसन लाइन बनाउन सक्दछौँ, जसले गर्दा बिरुवा क्रप फुड भनौँ त्यो फुडहरूको चाहिँ एग्रोनोमिकल ट्रेडलाई चाहिँ अपग्रेड गर्न सक्छौँ। डिजिज रेजिस्ट्यान्ट, ड्रट रेजिस्ट्यान्ट, त्यसपछि हाई इल्ड प्लान्टहरूलाई ग्रो गर्न सक्दछौँ। र अर्को भ्यालु एडेड फुडको डेभलप गर्न सक्छौँ हामी। जस्तै टमाटोमा भिटामिन डीलाई प्रड्युस गराउने, गोल्डन राइस त तपाईँहरूले सुन्नुभएको छ होला, गोल्डन राइसमा यसरी नै प्रयोग गरेर भिटामिन ए चाहिँ एडेड गरेको राइस हो। र अर्को चाहिँ रिसर्चको केसमा चाहिँ मैले जोड्न चाहेँ यहाँ। रिसर्चको केसमा चाहिँ जस्तै इकोलाई बेस्ड सिस्टम चाहिँ निकै सजिलो मेथड भएकोले मैले यता प्रस्तुत गरेँ। जस्तै हामी कहाँ चाहिँ अहिले पनि चाहिँ रिसर्च क्लोनिङ गर्नलाई चाहिँ इन्जाइम नहुने त्यस्ता प्रब्लमहरू छ, र इकोलाई सिस्टममा धेरै सजिलोसँग चाहिँ हामीले चाहिँ यो रिकम्बिनेन्ट डिएनए टेक्नोलोजी, प्रोटिन एक्सप्रेसन टेक्नोलोजी युज गरेर हामीलाई चाहिने जस्तै पिसिआरलाई चाहिने पोलिमरेज, रेस्ट्रिक्सन इन्जाइमहरू र त्यसै ट्रिप्सिन यस्ता चिजहरू हामीले इन हाउस प्रडक्सन गरेर आफ्नो रिसर्चलाई चाहिँ कन्टिन्यू गर्न सकिन्छ भन्ने मैले प्रस्तुत गर्न चाहेँ। यति भन्दै म आफ्नो प्रस्तुति टुङ्ग्याउन चाहन्छु, र यदि तपाईँहरूको क्वेसन छ भने भनौँ, पछि चाहिँ अहिले टाइम लिमिटको लागि चाहिँ म माफी माग्न चाहन्छु र धन्यवाद।
Thank you, Dr. Morgan, for your nice presentation on the emerging topics, that is plant vaccine and plant-based recombinant pharmaceuticals.
Rajesh Kumar, he is the student of master's degree from the agriculture and first year of agriculture and forestry university, Rampuchitavan. Please proceed your presentation.
Rajesh Kumar.
Hello. Am I audible, sir? Am I audible?
Yeah. Okay, you can proceed.
I'm problem with screen share.
And I'm sorry about
you can share the screen, but please do it
quickly. We don't have the time.
You will get just 5 minutes.
Yeah, 5 minutes.
Someone still screen. Share screen.
Dr. Morgan, please. I think this is your screen.
Yes.
Just a minute, please. I'm not seeing this screen here. एकछिन है त खै यहाँ मैले स्क्रिन अफ गर्न सक्छु। अफ भइरहेको छैन अहिले
भएको छैन। Dr. Ramari, could you please request IT department? I think they can minimize it.
I can share Rajesh PPT. Just wait.
Okay.
My PPT is slightly modified.
Oh, sorry.
So
you can. Can't you share? Try once, because you are allowed to share the file. So.
No. Still.
Rajesh को share हुन्छ होला त slide share गर्दा मिल्दैन?
मिल्छ, त्यहाँ share screen छ नि।
सर अझै मिलिरहेको छैन, स्क्रिन शेयर देखाइरहेको छ अझै। अघिल्लो स्पिकरको स्क्रिन हो सायद।
IT team, could you manage that, please?
Sure.
Okay.
Yeah, okay. This is the slide.
Good afternoon, everyone. I'm Rajesh Kumar, student of Agriculture Extension and Rural Sociology, Agriculture and Forestry University. First of all, I would like to thank NRNA for this beautiful opportunity And without any further delay, I would like to start my research findings and research details on the topic effect of different date of sowing and nitrogen level on production of direct seeded rice. This research was carried on 2017 in inner Tarai condition, Lomai Dang of Nepal, and the variety of rice is Savitri, and the situation is rainfed condition. The outline of presentation mainly covers the introduction, objective, materials and method, result and discussion, and finally the conclusion. Due to the time limit, I want to skip few slides. So rice is an important stable crop, and about 60% of the world population majorly depends on rice crop as a stable food, and rice ranks topmost crop in terms of area and the production with productivity 3.75 in Nepal. And the direct seeded rice is labor and water efficient and climate smart technology for the rice production. The sowing time of rice crop is important for two reasons. The first one is gets the appropriate level of temperature and solar radiation during vegetative period. And sowing on time guarantees that grain filling occurs with milder autumn's temperature, and it's more likely to achieve the best grain quality. And direct seeded rice is very much economical as compared to the transplanted rice, and it also matures early than the transplanted rice. And these are my objectives. The major theme of the objectives is determine the production and productivity of the rice with a different dosage of nitrogen fertilizer and time of sowing. This is my experimental details. The design I used for this research is 2 factorial RCVD. There are 3 replications with total plot size 520 meters square, and the individual plot size is 3 meter and 3 meter, and the space between each replication and each treatment is 0.5 meter respectively. The cropping history of the field is rice, wheat, and the fallow. And the research size I already mentioned. This research was conducted at Agronomy arm of Prithvi Technical College, which is conducted on June to September of 2017. And the average maximum and the minimum temperature during cropping season were 31.32 degrees Celsius and 23.32 degrees Celsius respectively. And the total rainfall during this research, I mean from June to September, is 2150.2 mm. The different factors, I already said 2 factorial RCVD. So there are 2 factors, days of planting, and the factor second is different doses of nitrogen. The first days of planting is June 5th, and the second one is June 20th, and the third is July 5th. And the different dose of nitrogen is the F1 symbols denote its control, in which we only use potash and the phosphorus with FIM, but in F2 and F3 and F4 we slightly increase the dose of nitrogen is 80-60-40 NPK, 100-60-40 NPK, and 120-60-40 NPK. And this is the combination that I use in experiment. Q1 means June 5th in control, and the different 2-well treatment are arranged. You can see in the screen, and this is my field layout. So the major result and discussion focus on 3 different topics: effect of different date of sowing on the plant height, yield and yield attributes of DSR, effect of different nitrogen level on the plant height, yield and yield attributes of DSR, and the interaction of the effect of a date of sowing and the nitrogen level on plant height, yield and yield attributing component of the direct seeded rice. The first is effect of different date of sowing. I would like to directly jump to the result. So
Dr. Rajesh Kumar, can you conclude? because we have no time. So
Okay. I will conclude within 2 minutes. So the best date of sowing is 20th June, which can be beneficial for farmer in terms of plant height, effective tiller, penicle length, and total grain yield. I mean, harvesting index will be very good at June 20th, and the fertilizer dose 120/60 is to 40 NPK is very effective in terms of nitrogen doses. And the interaction between sowing date and the nitrogen level significant with two interaction 20th June and 120/60 and 40 NPK nitrogen, potassium, phosphorus will be very effective in terms of inertiary condition and rain-fed condition. So I would like to conclude within a minute. Similar, I already mentioned the sowing date 20th June and the 120 kg nitrogen per hectare is very good for I observed in my research. So I would like to recommend for many investigation on this research.
Thank you, Rajesh Kumar. Please understand the situation. We have no time, so I would request for Sunil Mishra, please can just conclude your research, please. We have no time. Just share your screen and conclude your research.
Thank you so much, sir. Am I audible?
Yeah.
Okay. So namaste and good afternoon, everyone. Respected chair, coordinators.
Conclusion. Your topics and conclusion, please.
Okay. The prevalence of human Boca virus in children suspected with respiratory disease in Nepal. So directly moving to my conclusion. So in overall, prevalence of 12.5% was found to be in Nepal. And talking about male, so 15.58% was prevalent in male, and 96.98% of human Boca virus prevalence was observed in females out of 120 samples that were collected from Kanti Children's Hospital and processed at Central Diagnostic Laboratory by conducting RT-PCR, RT-multiplex PCR, to find the prevalence of human Boca virus, and this research has been conducted for the first time in Nepal. So highest cases below 2 years, and most of the patients who were visiting OPT department were found to be positive rather than the individuals who were admitted within the hospital. The month of March exhibited higher concentration of positive cases, and cough and fever were prevalence in all the positive cases, while wheezing was only present in 80% of the cases, and it leads to a conclusion that cough and fever are the outmost symptom of HBOV, while wheezing can lead to one of the major symptoms. Co-infection with parainfluenza virus was found to be 13.33%, and with RSV it was found to be 33.33, which seems that RSV and human Boca virus are mostly
Thank you, Sunil Mishra. We are short of time, so we have to close this session. And another really waiting, and they are using our room. So please,
thank you,
Dr. Balkumar, you will just conclude in 1 minute, and we will out of this session. We are so sorry, we are so kind. We could not ask any questions to
म्यामलाई धेरै धेरै धन्यवाद हजुरहरूको अमूल्य समयको बावजुद पनि यस प्रोग्राममा आइदिनु भयो आई एम भेरी लकी टु सेयर द स्क्रिन विथ यु अल एन्ड यहाँ आउनुभएका सम्पूर्ण स्पिकरहरूलाई पनि थ्याङ्क यू भन्न चाहन्छु र सरी टाइम लिमिटेसनले गर्दा तपाईँहरू सबैको प्रेजेन्टेसनहरू राम्रोसँग इन्क्लुड गर्न पनि सकेनौँ र क्वेसन एन्सर सेसन पनि गर्न असम्भव भएन यसको लागि वी आर भेरी सरी र थ्याङ्क यू सो मच फर your informative and nice presentation. Now I would like to end the session. Thank you so much.
Thank you, everyone. Thank you very much.
Thank you, everyone, for your nice.